But ARC was started 10 years ago, and what a decade of progress it has been. It's remarkable to see how far we've come. But the challenge for all of us in the room is this: how do we make the next 10 years even more impactful for patients? How do we make the next decade truly transformational? Again and again, we come back to one thing: data sharing. Without it, progress slows. We miss answers hidden in smaller populations. We duplicate efforts instead of accelerating them. And here's the truth: patients want this. They want their data to be used to drive discovery. They want their experiences to mean something, not to sit in a database, but to fuel the next breakthroughs. Patients are already sharing their stories, their time, and their trust with us. They expect us to do the same with each other. So if we're serious about being patient-centered, then sharing data is not optional. It is our responsibility. We need to start by openly discussing the incentives that stop individuals, institutions, and companies from sharing data, and find solutions that enable us to overcome these barriers. It's what partnership with patients demands of us. Because only then, when we share and partner, will we deliver the speed, precision, and the hope that patients deserve. And so there is no better segue. The Amyloidosis Forum is where the spirit of collaboration becomes action, where unique partnerships and scientific rigor come together to move the field forwards. This forum represents what's possible when every part of the ecosystem, every stakeholder commits to working together towards one goal: faster progress for patients. And with that, it's my pleasure to officially start the 2025 Amyloidosis Forum meeting and hand over to our wonderful Executive Director of Research, Kristen Sue.
Good morning, everyone. It's inspiring to see so many leaders from across the field gathered here today. Each of you brings a vital perspective to this work, and it's that diversity that makes the Amyloidosis Forum so impactful. When ARC launched this initiative in 2019, the goal was never to just host a meeting. It was to create a space where silos could be broken down, where collaboration is the norm, and where we can chart a collective path forward to advancing drug development in amyloidosis. And that mission continues today. The Amyloidosis Forum was built on a simple but powerful idea: progress requires partnership. It's a public-private partnership convened by ARC and is composed of regulatory agencies, industry, academia, and advocacy organizations, all working together to bridge gaps that slow progress. Our mission is to bring the greatest minds together to advance rigorous science and accelerate the development of therapies in AL and TTR amyloidosis. And it's not just a US effort, it's truly a global community involving experts from around the world. As Isabel mentioned, the forum functions as an ecosystem. ARC serves as the convener, but its strength comes from the way it unites regulators, clinicians, researchers, drug developers, and patient voices around shared priorities. This collaborative structure allows us to tackle complex questions, from trial endpoints and disease progression to real-world evidence and data sharing, as Isabel has declared, and move the science forward together. At its center of the forum is a steering committee of experts that guides our priorities and strategy, and it's supported by working groups focused on critical scientific and regulatory challenges that the community has prioritized. Now, the forum is more than an annual meeting. It's a continuous cycle of developing projects, refining definitions, conducting analyses, and publishing. It's not a single day of dialogue, but a year-round engine for progress, currently focused on these three critical areas that you'll hear about today: standardization of imaging, prognostic factors, and clinical trial endpoints. Now, none of this happens in isolation. I'll quote Preston Dunman, one of the founding members of the Amyloidosis Forum, by saying this slide represents an embarrassment of riches. We are so fortunate to benefit from the expertise, dedication, and generosity of so many united around these shared priorities. That collaboration has already driven meaningful change, and today's meeting is a continuation of that momentum, a chance to push the field further. Now, we'll start out this morning with some remarks and perspectives from Dr. Norman Stockbridge, followed by the sessions that will be exploring key challenges and innovative solutions shaping drug development in AL and TTR amyloidosis. We'll then move to discuss, hear from the projects that are being worked on through our 3 different working groups, starting with the imaging working group in the morning, and then the afternoon we'll turn to the prognostic factors working group and the endpoints working group. Now, before we get started, just a few logistics. We will have designated Q&A sessions during each moderated discussion, and we encourage you to participate. If you're here in person, please use the standing microphone at the center of the room. If you're joining us virtually, there will be individuals and experts monitoring the Q&A tool through Zoom. We've planned breaks throughout the day, and we ask that you help us stay on schedule by returning promptly after each one. For each of our speakers and moderators, there are timers, and please just be judicious in following them. Finally, I want to recognize the organizations and sponsors whose support makes this meeting possible, as well as the National Institute of Aging at NIH, which provided grant support for this forum. Their partnership sustains this vital work and, more importantly, ensures that patients remain at the center of everything we do. As always, I hope you'll keep 2 things in mind before we begin today's discussions. First, that the work that we're doing here is bigger than any single study, company, or program. And second, that by working together across sectors, across borders, and across disciplines, we have the power to fundamentally change the future for people living with amyloidosis. Thank you for being part of this and for everything that you do every day to push the field forward. I'm excited for the conversations ahead and the progress that will undoubtedly spark. So welcome to the Amyloidosis Forum. Now, to kick us off, it is a pleasure to welcome Dr. Norman Stockbridge, who recently retired as Director of the Division of Cardiology and Nephrology at FDA's Center for Drug Evaluation and Research. With over more than 2 decades at the agency, he's helped shape the modern regulatory landscape for cardiovascular therapies and was instrumental in advancing how we think about trial design, endpoints, and evidence generation. Dr. Stockbridge was also one of the earliest and most influential supporters of the Amyloidosis Forum, helping it grow into a vital platform for collaboration between regulators, researchers, clinicians, and industry. We're honored to have him here today with us to share his insights. Dr. Stockbridge.
Well, good morning. I'm going to strike on some of the same themes you've already heard. You people in this room this morning can appreciate how far we've come in these 10 years. Today, there's a very upbeat vibe in this room that could not be any more different than it was in those early days of marked by frustrations. How did this happen? As go numerous studies of developments for rare diseases, this journey starts with an individual unhappy with the lack of treatment options. This person elicits interest from others, patients, family members, caregivers, the occasional regulator. This community expands to include basic scientists and clinical researchers with an interest in their disease. And at this point, you've suddenly got an incubator in which medical product development can be nurtured. You've lowered the risk for a pharmaceutical company to get interested in a novel therapeutic area and perhaps help them identify effective interventions or benefits that might be demonstrable. people invest, and if you're fortunate, as you were in this case, success ensues. So everybody here should rejoice in the execution on this process of turning an unmet medical need into a thriving community, thinking about its next steps to improve on existing therapeutic options and broaden the coverage of patients. No one person does this. Each of you plays a vital role in it. We are living in what I think is the golden era of development efforts for rare and uncommon diseases, in part because such diseases are often pathophysiologically less complicated than many common diseases are, and in part because of the appropriate recognition by society, including regulators, that the evidentiary standards for both effectiveness and safety need not be the same in rare and common diseases. Like community engagement, these factors reduce the potential energy barrier for investments in rare diseases. But how these factors play out in terms of therapeutic options can be astonishingly different. In the case of cardiac amyloidosis, you got what you demanded, which was therapy that rigorously established benefits on, in this case, death and hospitalization. At the other end of the spectrum, consider the case of elimipritide for Barth syndrome, which is a much rarer entity, approved a few months ago with, and I encourage you to read the label, no reliable evidence of benefit for anything. The Barth community also got exactly what it demanded: hope. A vague hope unlikely ever to be turned into confirmed benefits. So thus the second thing I think this community should celebrate here is its commitment to deliver reliable evidence of clinical benefits. Your patients are better off for this, will forever be better off for this than the Barth patients will ever likely be. As you move into rarer forms of amyloid diseases, I trust that you'll continue this commitment. Thank you for the opportunity to join you today, and look forward to hearing how the next phases evolve. Thank you.
Thank you, Dr. Stockbridge. So we're now going to move into the first session of today's meeting focused on amyloidosis research at a crossroads, introducing the current challenges and then proposing some innovative solutions. And so I will invite our esteemed panel of discussants to come join us, for those of you who are here in person at the dais. This will be kicked off with a presentation from Dr. Matthew Maurer from New York Presbyterian Hospital, Columbia University Medical Center, followed by Ashutosh Wekelecker from University College of London, Royal Free London Foundation Trust. Thank you, Dr. Maurer. And for the rest, if you can, yes, come sit up here.
Thanks, everyone, for coming up. It's a pleasure to be with everybody. I want to echo the sentiments. It's really been an amazing journey over the last decade with everyone. Amazing progress and a lot of challenges ahead. I think we have enough seats for everybody. Yeah, better go there. Great. So I have the task of kicking us off about TTR, and Ashu will talk about AL. These are my disclosures. Thanks to everyone who supports our work, and I will talk a little bit about some investigational and novel treatments that are on the horizon. So just to take a moment and congratulate everyone. This is a slide we often show, but we've transitioned obviously from a disease that everyone thought was incredibly uncommon and very difficult to diagnose to one in which for a majority of patients we can diagnose on a single day without the need for a biopsy. It's quite common in various cardiovascular conditions. And enviably, we're now in clinic trying to explain to patients about different therapeutic options. And this has occurred in a relatively, I would say, short time period. So we've transitioned in this piece that I wrote with Pablo Garcia Pavia and Esther Gonzalez from TTR being, I think, the most frequent type. We thought there was kind of a single phenotype, but it's quite heterogeneous, and how are we going to tackle that? It's definitely not as rare as we thought. As I mentioned, non-invasive diagnosis is the norm in a vast majority of patients. I would say at least 85 or 90% at our center, and several treatment options are available. These are data from our center that one of our fellows, Nicholas Chan, put together years ago. I just highlight in the first bar all the way to the left, we saw between 2001 and 2013, 101 patients with cardiac amyloid, and they all had a biopsy in blue. And now we see 250 patients every year. So it's exploded, but between 2017 and 2021, there were 215 subjects. And as you can see, most were diagnosed without the need for a biopsy. And the staging systems, there are various ones. This uses the Columbia. We see that in gray, very few patients, thankfully, have advanced stage disease. And we are administering a lot more disease-modifying therapy. So earlier diagnosis and effective therapy leads to what is amazingly dramatic, I would say, improvements in outcomes. When I started doing amyloid patients, half of them were left the face of the earth in 2.5 years, and now we have median survivals that are probably at least 7 years, in my opinion, and potentially longer. I highlight this. I'm a heart failure doc, and our dots are the various trials have been conducted in HFpEF in general. I think this is a testament to precision medicine. You can see the number of patients enrolled in the trials and the absolute reductions in mortality. And none of these were statistically significant for mortality alone. And then there's ATTR ACT that stands out, as other trials in this space have been conducted, in which the absolute reduction in mortality was 13% with a small trial. And I credit actually our basic scientists for giving us the pathway to kind of achieve these goals in the clinical space. As you heard, we have multiple different therapies, and you'll hear more today. Stabilize just started us off, and we have tafamidis and acoramidis. We have ways to stop protein production hepatically with the various agents and maybe even gene editing. And I was somewhat skeptical. People know on this podium about amyloid removal, but I'm becoming more of a believer, though that's still experimental. And with all this progress, as we heard, we have a lot more to do. And these are questions that at least we encounter every day in clinic. Which therapy should we give the patient initially? Is there a particular therapeutic profile? Should we switch patients from one therapy to another, and why? Should we stop therapy? Do we need 2 therapies? Should we combine them? We're doing all of these decision-making in the absence of any head-to-head data or any even real-world data. We're hoping to treat asymptomatic patients, and as you know, there's a trial doing that right now. We're now allele carriers. And obviously, there's always concerns about long-term efficacy. We're very excited, but we haven't been doing this for that long. And I'll highlight there's still a huge financial burden to many patients. We as a community, now that we've had success, have transitioned into measuring disease progression. This is a table from a recent piece that many of us drafted, but there's no shortage of measures, either outcomes or other functional laboratory, PROs, EKG, imaging, measures of whether someone is doing well, God willing, regressing their disease or progressing their disease. And this has become a consternation for many of us. Should we be measuring this clinically? What do we do with this particular data, and how do we guide patients? I would also just caution us that just because we know a patient in our offices are progressing with any of these markers, what we don't know is that changing the TTR biological therapy is actually going to improve their outcomes. And I have some concerns about this because I show here this sigmoidal relationship that comes from kind of Alzheimer's work, where the brain can be super saturated at the end with amyloid, but patients can still progress. And there's really not much to target, if you will, the amyloid in that particular circumstance. So this is an area where I think we need some further investigation. And then, we have this classic paradigm in systolic heart failure where you have a myocardial infarction, a clinical event as outlined by Doug Mann. You get a reduced ejection fraction, and then you use commonly approved therapies that heart failure doctors use all the time, neurohormonal antagonists, and you can kind of reverse remodel the ventricle and improve things. And that same paradigm is now very exciting and emerging potentially with anti-amyloid therapy. Mariana and Julian and colleagues were the first, along with the seminal publication about claramitug in the New England Journal, to describe these 3 cases of patients in their center. Pretty rare phenomenon, 3 out of 1,663 controls who had marked improvements without any clinical intervention, any pharmacological intervention. This is one patient who you can see has an improvement in their EKG voltage. Here's another patient with multimodal imaging showing scintigraphy getting better and MRI getting better, both in regard to wall thickness and ECV. And here's a third patient who has EKG, echocardiographic, MRI, evidence of clinical improvement, and all of these patients developed polyclonal antibodies that targeted TTR and seemingly reversed their amyloid. And so we now, on the next horizon, have, I'd say, a whole host of clinical trials that are being conducted in the space of, if you will, anti-amyloid therapies. It's kind of hard to remember all the different companies that are involved and the different names. That could be a quiz. At least the first 2 up top did not meet their primary endpoint that was pre-specified. We've heard tantalating information from the sponsor that one of these agents, ansiolitamab, may have a clinically meaningful effect on a subgroup. We're waiting to hear more. And the other trials are all advancing quite rapidly in this particular space, and patients are quite enthusiastic with one trial fully enrolling like 13 months ahead of time. So this is a slide that John Spertus had developed talking about the KCCQ, and I borrowed it from his piece, but I get a little concerned about how we design clinical trials, and I know we pick a primary endpoint and want to know, do we meet the broad effect for the patients that we studied? But I think he nicely highlights here that we usually have a pretty heterogeneous group of patients, and one could argue in the world of rare diseases, and especially amyloid that's multisystemic, that heterogeneity is more than the norm. And we look at the, if you will, mean treatment effect in quote unquote the average patient who's not really representative of anything going on. And so it really, that effect doesn't really represent the outcome in any individual patient truly in the trial. And, maybe there are ways by describing, if you will, the proportion of patients with different clinical effects shown in panel B, we can better determine what's going on. And I'd be very interested in my colleagues' input and that of regulators. Is there a better way, especially for rare diseases, to design our approaches? I think we have to be a little bit weary. We're very able to, I think, stop the production of hepatic TTR, but we all should be reminded that transthyretin is produced in the choroid plexus. These are data from our Japanese colleagues in patients who had liver transplantations in the age of 30 or 35, and 15 years later had demonstrable PET scans showing amyloid in their brain. And so this is a phenotype similar to ocular manifestations that I think patients care a tremendous amount about, and I don't know what we're doing for them. I don't know if there's a solution, but I would highlight that some of these agents may cross the blood-brain barrier and might have some efficacy, but we just don't have any sense of that. And then I would say, finally, having just come from clinic yesterday, patients obviously are thrilled with our outcomes and what we've been doing, but they remain very concerned about the access to treatments. I saw a woman yesterday who we've tried now for 6 months to get her access to a treatment. She has wild type TTR amyloid. She has an insurer that doesn't have the medication on formulary, and we advised her to just enroll in straight Medicare yesterday, but it's somewhat problematic as a clinician and a patient when you have treatments and you can't bring them to bear. And these data actually, congrats to ARC for collecting them, used a comprehensive score for financial toxicity called COST that's been used in the oncologic literature. And you can see that financial toxicity and concerns from patients is the norm. And so anything we can do to ultimately ensure widespread access, not just in the US, but internationally, I think will be incredibly important. So in summary, I would say that thanks to our basic science colleagues, they have helped us develop unbelievably effective therapies. They all work, they're all effective. And I think the key right now for us clinically is to make sure we're getting patients access to them and continued access in a timely manner. We are, as clinicians and patients, I think in this very enviable position of trying to choose among what therapy to give someone, but I have no idea how to design or figure that out, and that's very frustrating to patients. And then finally, I think that the ongoing trials and maybe, as we heard about, more data access and comparative effectiveness studies might provide us with important insights into how to best manage our patients. So thanks for your time and attention, and I'll turn it over to Ashu.
Thank you, Matt, for that amazing presentation on ATTR amyloidosis, and also to Isabel, Kristen, and the team for organizing this forum and inviting me here. So I'm going to talk about AL amyloidosis. Where are we and what are the unresolved issues? And this talk is going to be more on what we need to do in AL. There are advances, and there are really good advances in AL. But the landscape is not as simple as it was in ATTR. AL is rare, and it'll always remain rare, between 8 to 10 patients per million. As patients go beyond frontline treatment, the disease becomes rarer because some patients will die, some patients don't relapse for a long time. We have had a number of therapies that have been adopted from multiple myeloma field in an off-license label, and that has changed our targets for therapy. That have changed what we might define as success of treatment. And in a rare disease area where we have a disease which has got two different things going on at the same time, or rather three different things going on at the same time. We've got the plasma cell clone, which is producing your abnormal protein. We have got the abnormal monoclonal protein itself, which is causing significant toxicity. And then we have got the fibrillary deposits directly causing end organ dysfunction. We are trying to address three separate endpoints for AL amyloidosis, and we need to be able to design trials which can address these issues. Our success has mainly been in the area of the plasma cell clone, and we still remain to see what happens in the other areas. I picked this slide of Matt, and I thought this really well illustrated the problem of AL, and the patient population is heterogeneous. But to me, this slide, if we use it for TTR as an example, is actually very simple. AL is very different. We first have two different types of light chains, AL kappa and AL lambda. These patients are different. The fibrils in each individual patient are unique. So each patient's fibril structure is different. Then they have an underlying plasma cell clone. This can start from 1% to 5% to 10% to true multiple myeloma for more than 90%, and those patients will behave differently. We then come to the issue of which organs are involved. It's not just a cardiac disease. You have multi-organ involvement, cardiac, renal being the commonest, but also autonomic, GI, peripheral nerve, all of which don't really have well-defined endpoints. And it's very hard to have a clear endpoint for these patients, and they can be in any combination. Patients can have heart and nerve, heart and kidney, liver and nerve, and that makes it much more difficult. So when we have got a single endpoint and a trial fails, well, not really surprising. On the other hand, if we do a single intervention, we may find that actually we are looking at 3 or 4 different patient populations. And your trial might work in a subgroup, as we tantalizingly heard from Matt earlier, but might not work in any other subgroups. And we need to be able to have trial designs which can adapt and have that flexibility of using those subgroups to define our success. And if we don't do that, we will never be able to progress in AL trials. So what's happened? We're very jealous of all our TTR colleagues. You've got lots of licensed drugs. We have had lots of clinical trials. These are about 100 trials going on in AL amyloidosis globally right now. Most of these are phase 2 trials. But we've only had one frontline phase 3 randomized control trial. We have got 2 phase 3 trials for imaging amyloid, which we hope will read out very soon. We have had upfront trials of antifibril antibodies, and we'll touch on that. And we've only had 1 randomized phase 3 trial in relapse disease, which never went anywhere. And we've only got 1 licensed treatment for AL, which is DARA-VCD. So over the last 25 years, we've had a lot of success, but we haven't had a lot of clinical trials. And the treatment success has mainly been driven by treatment of the underlying plasma cell dyscrasia. How is the success being defined? The patient survival has improved enormously. When I started in amyloid nearly 20 years ago now, the patient survival was about 2 to 2.5 years. And the data on the left-hand side are from the Boston University Medical Center, from the right-hand side from our own patient cohort. Median survival has very clearly gone up from 2 years to 5 years to 7 years, and our latest cohort, it's even longer than 5 years. So whatever we have done has clearly worked. Our patients are living longer. And as I'm always reminded, living longer and feeling better are the 2 things we are looking for in licensing a drug. There is improvement in their quality of life as we are achieving better and better responses. We started off with alkylator-based treatments, and this was until 2012. Patients, when they were treated with alkylators, our complete response rates were about 10%, 12%, and majority of the patients' responses were partial responses. We then got bortezomib-based therapies. Bortezomib is specifically targeting plasma cells in amyloid, and our response rates went up to about 60-70% with very good partial responses seen in about 50 to 60% of the patients. The improving response rates meant that we had to redefine our response criteria. We had to come up with a new category of response, which is very good partial response and a complete response category, which was not seen that frequently in previous therapies. And clearly, we were able to show that for each category of response, the outcomes were better. We then had daratumumab-based treatment, where the complete response rates are now 50 to 60%. But we also realized that the free light chains dropped so much that our previous response criteria did not capture the response well. So we had to go back and look at the data and issue a clarification of the response criteria, showing that the free light chain ratio was no longer accurate in defining a complete response. We had to actually look at the light chain value, and the outcomes were exactly the same. So we've got good success, but that has also made us redefine what we call success, particularly from a regulatory perspective. Our expectations have changed. With DARA-based treatments, the responses are very rapid. With alkylator-based treatments, it would take 4 or 5 months for response. With bortezomib, 3 months for response. With daratumumab-based treatments, we are seeing responses within a month and best responses within 3 or 4 months. And this also means that patients are living longer, but we also need to change what we do from a response assessment perspective. These deep and rapid responses, certainly in the final analysis of the Andromeda clinical trial, have shown that 75% of these patients are alive at 5 years, and a vast majority of these patients in a complete response would be progression-free at this time, about 60%. So clearly, a lot of patients are living longer, but this great success means that overall survival becomes a challenging endpoint for AL clinical trials, even in the newly diagnosed setting with a DARA-based regimen. But we also know that all patients will relapse eventually, or most patients will relapse, we think. And when we looked at, this is a cohort of about 1,800 patients from the Amyloid Center, and this is looking at first, second, third, fourth relapse. There is mortality at every single time the patient relapses, and that mortality is very similar to the mortality we see in the frontline setting. However, when we get these patients into a complete response, they still survive longer. So CR in relapse still translates to better outcomes. We need to treat these patients early after relapse is one of the lessons that we've learned. Very low levels of light chains can be quite tissue toxic, and we need to define progression criteria we'll hear about in the future discussions. And then CR still remains a goal even in the relapsed refractory setting. By using our best drugs in the frontline setting, we've created ourselves an unmet need. What do we do when patients don't respond to DARA-VCD or patients relapse after DARA-VCD because all our best agents, anti-CD38, bortezomib, is being used in the frontline setting? And about 40% patients will not get a CR, and patients are progressing now a few years after DARA-VCD. We have an ever-expanding range of targets that we can use for plasma cell therapies in AL amyloidosis. This is taken from our myeloma literature, targeting BCMA, other plasma cell antigens, bispecific antibodies, CAR T-cell therapies, small molecule inhibitors, and drug-conjugated antibodies. And all of this could be potentially used for AL treatment. One of the specific areas where AL could potentially be an area for study is T1114. 50% of AL patients have T1114. 70% of patients with a light chain excess and no M protein have a T1114 translocation. The responses with BCL2 inhibition are very good in this setting. Overall response rates of about 70%, with complete response rates of 70% when we look at the T1114 population as well tolerated. We have a number of phase 2 clinical trials that are going on with venetoclax, but we don't have a licensing study with this agent, despite us having known this fact for nearly 15 years. BCL2 targeting treatments will be effective in AL, both in frontline and relapse setting, but we need to study and prove this point. BCMA is an antigen on plasma cells, and targeting BCMA has become very important in multiple myeloma. This is the review that we wrote, just summarizing various BCMA-directed therapies in AL amyloidosis. And as you can see, the response rates using either antibody-drug conjugate, belantamab, bispecific antibodies, teclistamab, or CAR T-cell therapy, we are seeing responses in the order of 90 to 95%, with complete responses of 70 to 80%. And this is much different to what we've been used to seeing in the relapse setting, and certainly even better than what we've seen in the newly diagnosed setting. And I'll just present to you one slide showing the results, and this is from data presented at the recent ASCO meeting of a BCMA-directed CAR T-cell treatment, where patients, when they were treated, you can see that the free light chain levels drop back to normal literally within a week of starting therapy. And this very rapid response rate is critical, hopefully, for improving patient outcomes. So I come back to my graph again. Our goals have changed a bit further. We are now expecting response rates, once we have started using BCMA-directed therapies, of 90 to 100%. What we're also expecting is that these response rates are very quick, often within a week or 2 weeks of starting therapy. It has raised some challenges. The light chains become undetectable. So our current response criteria will not be able to capture this because the light chain ratio is no longer calculable. These patients are in the CR, but they still have an intact monoclonal protein, which will last for many months. And therefore, we won't capture the true benefits of these treatments if we just use our traditional response criteria. And we need to redefine what success is in this era of the very novel therapies for amyloidosis. But there is a lot more to come. We have got antibodies directed against 2 antigens, trispecific antibodies. There is a cohort of patients that will hopefully be studied in the Janssen BCMA GPRC trispecific antibody. We've got 2 studies with CD19 BCMA-directed CAR T-cells that we are opening in our center very soon. There is data about in vitro CAR T-cells, and amyloidosis is a perfectly ripe population for this because they have a very low plasma cell burden. You could simply give them the target vector, produce the CAR T-cell in vitro. You don't need any bridging therapies. And finally, we could use macrophages to help us to clear amyloid deposits rapidly. But moving beyond light chain therapies, we've just heard from Matt about light chain stabilizers improving outcomes in ATTR. We've never had anything like this in AL, but in theory could be much more effective because the light chains in AL are tissue toxic in addition to forming amyloid themselves. And we hopefully have a small molecule which will go into clinical trials in the near future, looking for stabilization in amyloidosis. But one of the important things is removing amyloid. We know that natural clearance is slow. When we look at overall response rates in patients with AL amyloidosis, particularly cardiac response rates, the responses improve over time. A third of patients have a cardiac complete response at 2 years, at 1 year going up to 50% at 2 years. But this means that there are deaths at every stage. 25% of the patients will die within 2 years for stage 2 disease and stage 3, and 50% in 3B. Admittedly, these are patients who are not treated in the daratumumab era, and therefore there is a critical need to have a fibril removing agent or an antifibril strategy for AL amyloidosis. We saw this from Matt before, that we reported 4 patients from the National Amyloidosis Center that amyloid can be removed by naturally occurring antibodies. Their hearts do improve. So the concept in these naturally occurring antibodies has been proven. We just need to work out how do we prove it in our own patient population, and it's been disappointing in AL. We did a trial of anti-SAP, antifibril antibody. There's a paper which I suspect none of you have ever seen because the study never made it to light. We then had some disappointing results from the NEOD-001 bertamimab, and now we have some intriguing results from the KL-101 antibody program where the primary endpoint wasn't met, but there's a suggestion that maybe it'll benefit a subgroup of patients. And I'm not very surprised to hear that. We know that amyloid regression in individual organs is very different. Amyloid can regress very quickly from the liver and spleen, but may regress more slowly from the heart. But that also means the accumulation can be different. So even if you have a very slight relapse, the heart will get much worse before any of the organs get worse. And all of that needs to be captured in an antifibril antibody-based strategy. So what do we need for amyloid? Yes, we have had a lot of success with antiplasma cell therapy, much of it being off-label use of drugs which are licensed for myeloma. We need to remember that in this rare disease setting, we need to have novel endpoints for clinical trials. There is always going to be a small sample size. We are always going to have to use some form of composite endpoints. It may be a mixture of response rates and organ response, maybe overall response. We need to remember that it's not just the physical protein deposit or the light chains, but there is proteotoxicity. So we need to be able to use biomarkers of organ toxicity for capturing drugs which can stabilize the amyloid fibrils. We need to be able to use imaging to tell us that the amyloid is getting better, and we should have imaging endpoints which allow us to track where the amyloid is, either using MRI or GLS or maybe amyloid-specific antibodies. Because AL patients are always going to be heterogeneous with many organ involvement, we will need some form of hierarchical endpoints. Maybe cardiac, maybe renal, maybe a composite mixture, maybe quality of life, which we all know is so important for these patients, and we have never had a trial with quality of life endpoint. And potentially with these very new treatments, we might be able to capture this much better than we did with traditional old-fashioned chemotherapy. And the biggest challenge, of course, is how do we validate these endpoints in this rare population? I just showed you data from 2,000 patients in one study, 1,500 patients in another study, and about 5,000 patients in a recent retrospective study. We will need to use these cohorts of patients from large centers and registries to define our endpoints that we've used for clinical trials, and using them as comparators may not be perfect, but still good enough to tell us that we are doing better and we can use these endpoints for getting these drugs licensed. And then there are some precedents that have been there before, but each individual study will need to be designed separately and allow us to have the flexibility to get these outcomes. Thank you all very much.
Thanks, Ashu. That was super. And I think we're going to turn now to our panelists. Hopefully this will be interactive. Anyone can chime in, but I'll start off maybe with Dr. Masri. We heard a lot about, data sharing and the needs, I think, of our population to better understand in clinic how to utilize these therapies. Any thoughts on your part about the role of real-world data registries and how we can, as a community, come together more seamlessly in this particular regard?
Thank you for the question. I think it's not easy, but having multiple approved therapies is a good thing. We get, not every patient responds to everything. Not every patient tolerates every therapy, even if the therapy is approved. The challenge that we're facing, as you nicely highlighted, is what to do in the clinic when someone asks, well, which drug should I go on and which drug is best suited for me? And I think, really honestly, I don't think the clinical trials that led to approval of these drugs are helpful in trying to understand which drug works better. And it will only be on us as a community to try and understand in the real world the performance of these drugs. The challenge is that there is inherent selection bias. Who wants to go on an oral therapy, who wants to go on an injectable, who wants to do this, who wants to do that. And so those are some of the challenges. Can we actually run a study that compares these different patients who end up going on different commercial therapies? I think that's possible. And I think that this is an initiative that we should do. How do you incorporate that into future clinical trial design? I think that's the more challenging question. And, when ATTRACT started, there were no approved therapies and it was against placebo. But now we're 10 plus years later and we're still designing trials also against placebo. So that's something to also consider and take into account as we evolve over the next decade in TTR therapies.
May I pick up on part of your question, Matt, about, I think there's a piece about data sharing embedded in there. And of course, Isabel, I think, spoke eloquently about that. I just wanted to emphasize there's many barriers to data sharing that are understandable. There's also many ways to make it happen, but what's necessary is that open discussion that Isabel was talking about. There's approaches to having data use agreements that protect the data holder. There's federated approaches so the data holder can still govern the data the way they need to. But to kind of find those paths, which can sometimes be complicated and take some creativity, that open discussion of what the barriers are is so important. I think for a lot of these questions, especially like combination therapy, sequential therapy, it'll be great if we have clinical trials that answer those, but that'll probably take a while. A lot of the best answers are going to come from real world data, and the faster we can like connect the right data, questions, analytics, the faster we're going to get answers.
Yeah, I wonder if maybe our colleagues from FDA, Jenny, Dr. Lee, and from AHA could comment further, because you guys have a depth and breadth of experience that we don't have in navigating this and garnering, obviously, high quality data that can best inform the community. So suggestions or thoughts in this particular regard as we try to move forward.
Yeah, so FDA support treatment development for rare diseases through many programs and initiatives. Last year, there was a public workshop bring together stakeholders to discuss the use of natural history disease data and the registry data for drug development. I thought the meeting was really informative. Perhaps here I will reiterate some takeaway message and some lessons learned in terms of using those real world data. We heard challenges across facing rare disease drug development program, either lack of this natural history disease data or registry data, or data do exist, but they exist in silos. A third challenge, the data may not be collected or managed that meet a regulatory standard. So facing those challenges, there's several lessons learned. One is, so patient registry and nature disease data could be fundamental, very useful tool for rare disease treatment. Try to standardize a data collection, use established data elements. Third, always assume that the data you collect from the beginning may someday be used for regulatory purpose. So safeguard your raw data. You may be used multiple times in future. Or use a state-of-art data management system, so you ensure quality, including the procedures, processes, make sure it meets a regulatory standard. Those data are invaluable in terms of drug development for rare disease. However, using those data for regulatory purpose does increase the complexity in terms of regulatory decision-making when sponsors submit data. So that's require early involvement with FDA. That's require your data reliable and relevant, and has to be auditable when it serves a regulatory purpose. So perhaps make sure you access to patient level data, not only summary level or population level results. So this is some thoughts I thought maybe I'll share here.
Very helpful. Christine, anything?
Yeah, so registries should improve patient outcomes, period. That should be their purpose. If we don't think we can improve patient outcomes, we shouldn't engage in registries. Unfortunately, we know that while we all carry that torch and will continue to carry that torch, altruism is not what drives the US healthcare system. So I think in addition to the nuances of operationalizing a registry that's of high enough quality that could be used for regulatory purposes, we need to think about drivers within the healthcare system that will incent data sharing. And I think part of that is recognizing hospitals for sharing their data. Part of that is data harmonization, so we have more power, we can have more findings, and that benefits companies. And I think part of that is really investing in creating systems of care and referral networks. So there is a differentiation by utilization of registries and designations or certifications for when providers are sending their sites to a center that they know that they're receiving high quality care. And as we start to collaborate to build a registry, harmonization is obviously important, but as Jenny said, we have to make sure that it's fit for purpose. As we have more therapies, trials are going to become more complex, and it's going to be really important to have a high quality real world data set that could really stand up to an FDA audit were we to use it for a control arm. So I think all of these things are really important, and all of us need to be at the table as we vision this future to make sure that we're building the right mechanism to really solve this issue.
Yeah, I totally agree. And obviously, we've seen the landscape change so dramatically that I think if we are fortunate enough to use real world data for a control arm, it's going to have to be contemporary, because I think in these fields, historical data, as we learned. Mitch, you've written a lot about AI, and one of the things that I think we waste a tremendous amount of time when we have a large registry at our institution is, copying data from one data source into another. I'm sure there are many other things that AI could do for us, but you've given a lot of thought about this. Where does AI or these machine learning approaches in trying to harmonize data help us in this regard?
Thanks. So I want to just make a brief side note that what I'm going to discuss comes from a public-private partnership, which is called the Heart Failure Collaboratory, which is tasked and really a multi-stakeholder organization that's goal is to increase efficiency of clinical evidence generation. And we've done a lot of thinking about current and future uses of artificial intelligence. I think, you highlight, Matt, one of the great utilizations of these current AI tools that exist, which is really to take over manual labor activities that really require a lot of personnel power and time right now, and can do them much quicker. And I think that those are current opportunities. I think AI, what's interesting is it's currently a space where the degree of investment and the future potential still far outstrips its current applications in a validated sense. I think that if you look throughout the clinical trial apparatus, there are potentially many use cases for AI to make clinical trials more efficient, to make clinical evidence generation more efficient. But I think probably where there is greatest evidence of both individual institutions implementing this in their own electronic health records, but also multiple companies trying to really validate this approach is in patient identification, clinical trial enrichment, and in limiting of heterogeneity of patients to try to improve your ability to demonstrate an effect, a treatment effect in a specific population. Probably less so in the sort of prediction of a clinical trial endpoint, but definitely in the identification of patients who, in terms of prognostic enrichment, are at highest risk for having an event, thus allowing you to shrink a clinical trial in terms of overall enrollment and really be most efficient in the way that you're taking that forward. So I think that that's where AI can be most harnessed, particularly in this field, and I think in both disease states that we've talked about, can really be leveraged to improve the overall efficiency the apparatus. There are lots of other ways that you could think about doing this, but I think in general, those are not necessarily ready for prime time. If I can just comment on an adjacent topic, which is this issue of comparative effectiveness of treatments that currently exist. The AI is not necessarily going to be the solution for determining which therapy to give first, but the digital health apparatus that currently exists has shown that you can really create a pragmatic clinical trial based within electronic health records relatively cheaply for interventions that really have a well-determined safety profile. And you're just looking at major events that could be reasonably captured within an electronic health record or in the basis of clinical care. And so while it may not inform regulatory decision-making, it could certainly inform clinical decision-making. And I would suggest that this sort of pragmatic, digital-based comparative effectiveness study could be implemented at a lot of centers and utilize perhaps AI to identify patients, because we know that there are many regions of the country where patients with amyloid are not being well found. There's a huge opportunity to find more of them. And you could do such a trial to really answer the clinical question of what we all should be doing with our patients in terms of sequencing or adding on therapies that may not inform regulatory decision-making, but that's probably okay.
Thank you. I'm going to follow in the same vein, maybe this time turn more to hematology. So maybe starting with, go to Angela and Vaishali. Again, for AL patients, we have our own individual registries, which are looking backwards quite a lot. What do we think we should do in terms of making that our data is contemporaneous, as was said before, in terms of not looking at MDEX-treated patients and survival benefits, which we can report very nicely, but the new patients will never see that so quickly because we are seeing changing outcomes. So I don't know whether you have any comments on that, and then maybe what we've done with our antifibril strategies and how we can refine that a bit further.
Okay, so I'll go first. I think in the AL space, we've been pretty good about collaborating and putting our registries together. The major issue, however, is what we're collecting is not what regulatory bodies want to see. So back in the day when patients lived a year or 3 years or 5 years, you could get the baseline data and then say, okay, we have a survival difference. But when it comes to registration studies, what I've learned so far from FDA, and please correct me, is functional endpoints are what are key. If you can't use, overall survival, is it a progression-free survival? Is it a some kind of PRO? Like, what is it? And I think to this point, we have been collecting all kinds of information. And I think the ATTR space is going to experience this more too, because as patients live longer, hospitalizations and death just isn't going to fully cut it, because fortunately, our patients are living longer. And so, we become victims of our own success. And so I think really understanding what needs to go in these registries is as important as harmonizing, the baseline elements and, the treatments and all the rest. And so, like, you've done amazing work with patient-reported outcomes and that kind of thing, that next phase of what we need to collect to really have the right data to then use as a comparator.
Thank you.
So thank you, Ashu, for that question. I think data sharing, as Angela said, is critical. And I think data harmonization, what data fields to collect and what data fields to add on, because we all have our databases and we all have our repositories and we all have our longitudinal, institution-wise. But, we were not really looking at NT-proBNP in 1990. And we've started looking at that since 2000s. And when do we really add it on? Do we really do the NT-proBNPs on frozen sera from 1990s to really have a longitudinal assessment over a long period of time for a particular biomarker? We were not really collecting for biorepositories, we were not collecting plasma in 1990s. We've started doing that in 2014. So again, I think some harmonization across all centers, what to collect, what data fields to collect, what PROs to collect, I think is absolutely critical for this to be really shared across centers, as well as, that it meets the regulatory requirements, as well as the data can be auditable at some point. I think the other thing that I kind of wrote down as you were presenting your stuff, which was very nicely done, is, Matt mentioned about the financial toxicities in ATTR drugs. Financial toxicity in AL amyloidosis, it's not just DARA and bortezomib. It is, these bispecifics are unbelievably expensive. CAR-Ts are unbelievably expensive. And most of the payers in the United States are not paying for bispecific antibodies for relapsed AL amyloidosis because it is not yet FDA approved. It is approved for multiple myeloma. But because it is so expensive that they are looking at the data and the records more carefully and not just approving it. So I think financial toxicity of these novel agents, which are extremely effective, is going to be a major challenge, that which payers are going to approve it, which payers are going to pay for it. And I think that is one other thing that I think is going to be in future, we are going to be discussing that even more. I think one other thing that I wrote down in your novel endpoint solution, you didn't really have the major organ deterioration progression-free survival, which was really a secondary endpoint for Andromeda clinical trial that led to approval of DARA's VCD in frontline setting. And that really endpoint was not really truly validated in any setting, but regulatory agencies did take that into account or did really approve it based on that MOD PFS. So I do want to kind of ask or discuss that issue as well. And I think as the therapies are becoming more and more effective and DARA VCD becomes the standard of care, what is going to be our control arm for any clinical trial in AL? Your hematologic response is the only endpoint that is really has been validated to improve survival, or it is a surrogate for survival. But now, your hematologic response rates are 100%. So what is going to be a control arm that is going to really, so I think there are a lot of questions in AL as they are in ATTR, and I think these questions are, and I think I remain in business because these questions really excite me still. So I don't know whether I answered your question, but I did have a lot of questions that I wrote down. So
thank you, Vaishali, and Angela, for touching on those points. I might come back to Christine and Jenny, if I might do in that vein, because clearly, as Angela said, our survival is so good now that survival endpoints are unlikely to be there. Progression-free survival is prolonged. 60% patients haven't progressed at 5 years. That creates a challenge in PFS. And we've got a lot of biomarkers which we know change very early, which will really make it helpful for us to assess how these patients are responding. And then the point which Vaishali made, that in the Andromeda trial, MOD PFS was used as an endpoint, but had never been validated in a clinical trial. The complete response criteria in Andromeda was also never validated or reported anywhere. And one of the slides I showed was the clarification of the response criteria was in relationship to discussions with the FDA at the time the Andromeda trial was actually being analyzed. So how can we work with regulators to get these new endpoints and figure out how we do it? Because we'll be working along with the clinical trials running at the same time. and the data will keep on accumulating. So any suggestions, any guidance would be very valuable.
Well, I will try. Those are good questions. Not mean to provide assurance. This is actually not unique to AL. Other rare diseases facing similar challenge. You have a 0 approved product, but today you have 1 or 2 already went to market. It seems like trials are not your only option now, because you're no longer in pre-approval setting. You only you were in a data lacking environment in pre-approval setting. Now you have an approved product in the market. Patients are using it. Some are using it, some are not using it. Perhaps consider looking into real-world data that collecting from routine care, EHR data, electronic healthcare data, or insurance claims data, or registry, or now the technology allow us to link them together to get an even more enriched data environment. You have a longitudinal, you can follow patient from day 1, they start treatment, till the health outcome that you want to capture. So you seem like you have more option in the post-marketing section. The second point, once a product approved, you can really collect data first on how patients use those products, what's their patient disease characteristic, how's the treatment effect. And those data from product 1 may in future be useful for second product when they're under assessment as a new baseline. If the survival prolonged, if the disease progress changed, improved, so that's options available.
And I'll just add that I think that registries do hold an opportunity for us to engage, not even necessarily in phase 4 studies, but even some of these validation of some of these surrogate endpoints in a very pragmatic way. I think with more patients in the real world, we have more data. We have an opportunity to look at a whole host of surrogate endpoints. And I think what we don't always consider are what endpoints are important to patients. We think about regulatory endpoints, but if you ask a patient, that Venn diagram doesn't always overlap between what's really important for patients and what's important for regulators. And I think we can't lose that in the conversation. We have to continue to push the envelope to say, these functional outcomes are what patients are prioritizing. This is really important to them. Their quality of life is important to them. So how do we make the case for that as well? And I think I'd just finally say that long-term follow-up is key. And I think we're seeing patients are surviving many more years, which is wonderful. And I think that adds complexity to what we can understand in terms of outcomes. And I agree, I think we may be setting new bars for what we would expect to see in survival. So surrogate endpoints are going to become increasingly important, but we have to have an infrastructure to follow patients long-term because we need to understand the effects of these therapies long-term.
May I chime in on this one?
Of course.
I don't want to be the broken record, but if I must. So I think there's a question about clinical trials are ongoing, and at the same time, we have to figure out endpoints. I think the registries are definitely important for that. I think learning from the recently completed clinical trials is just an incredibly valuable resource. As Vaishali was saying, sometimes the registries were not yet collecting all the good stuff. A lot of times the trials have that. It may not be their primary endpoint or secondary, but they usually have very rigorous collection of the type of information that we would need to look at carefully to learn about endpoints. So I think, again, the faster we can share or give analytic access to recently completed clinical trials so the community can learn from it, that's a really important part of improving the next generation of trials and how we look at their endpoints.
Yeah, Oh, go ahead, Angela.
No, I just, like, not to pull out names, but, the Prothena data, we've never, like, a lot of data just isn't out there. And what Isabel said, I mean, patients in good faith went on studies and, to really get some of those data shared, it would be so, so valuable. And I can't say enough about that.
And also the placebo data, because most of these trials were, placebo-controlled trial.
And
So
and the
though
re-
that is critically
Right,
important.
and again, I don't even care what arm the patient is on, you could say A or B, it doesn't matter, but just to have that really systematic collection gives us such an opportunity to validate things and to understand things and to take us to the next level.
Yeah, go ahead.
Just a quick point about the surrogates. I mean, we do spend time looking at surrogates in the real world, and we do spend time connecting that to outcomes. One of the issues is once you have a precedent, there is also the side of the sponsor of these new drugs. They usually don't want to go for surrogates themselves. And so, we can talk about this, we can work on it for sure, but also we need partners, those who hold the keys to these new investigational products. For example, take the example of TTR. We now know that TTR silencing is a strategy that likely mediates the outcomes on these trials. But every subsequent trial will likely still look at outcomes so that they actually can be compared to the previous treatments or can be used in the same light. So I think that's another challenge to those surrogates to be used as a temporary, accelerated approval, for example, or a surrogate endpoint.
Yeah, I would echo a lot of the sentiments. I mean, I think obviously we all conduct clinical research under the guise of, regulations and the FDA, and I think we all emphasize that data is really fantastic. It's much better than any of the data we collect, so we want to encourage all of those who are engaged in this to try to share data. We could, combine, analyze those trials and really figure out whether there is a valid surrogate across mechanisms of disease and bring that data back to regulators that might actually shorten the time frame. I do echo Norm's sentiments earlier, though. I think we as a community have done a great job at really giving robust evidence to our patients that allow us to really tell them, in clinic what they're able to achieve with these therapies. I did want to encourage anyone who has questions or comments to please go to the microphone as well. And I wanted to just express my envy. In our work, obviously, with oncologists, and they seem to utilize imaging a lot to determine what goes on in an individual patient. You have a tumor somewhere, and the tumor is getting smaller, and that's a marker of efficacy. I'm no expert in regulatory requirements, but maybe Sharmila and Spencer, with the advent of multimodal imaging and being able to maybe visualize amyloid, quantify it, maybe you guys can comment a little bit, and then we can go to the questions.
Okay, I can go first. Thank you to both of you actually for amazing presentations and such a great discussion. So what stood out for me from both the talks is the comment about patient heterogeneity. And I think that is where imaging could play a role. Amyloidosis is a very complex disease, as all of you know. There's a component of the precursor protein, there's a component of fibril, and then there's a component of end organ damage and dysfunction. So right now we're trying to give a standard therapy for the whole group and trying to hope that everybody improves. But of course, how a given individual responds to a given amount of amyloid in their heart or kidney is very different, likely, and it depends on maybe their immune response to the disease and their overall disease status. The biomarkers that we're using in clinical practice that both of you alluded to are actually excellent, and I know that patient outcomes are very important for patients, for us, and for the regulators. Of course, everything goes well as long as the drugs are improving patient outcomes. But the challenge is, if the drug does not improve a patient outcome, does it mean the drug is not working? I think that is where imaging and all these advanced ways of looking at disease heterogeneity could play a very important role. So the question is, any given degree of heart failure or heart failure progression, right, Is it from amyloid, or maybe it's the amyloid is well controlled and the treatment is working well on the amyloid, but the patient has ongoing heart failure because the heart is beyond repair, so to say. And at this point, we can't differentiate the two entities. So this is where I believe imaging can have an important role in quantifying or separating out what is amyloid in any given patient from what is the effect of amyloid on heart failure or organ disease and organ dysfunction. And I hope that being more specific about the pathology that we are targeting can improve outcomes in these patients. Currently, we don't have that, and I think that is where we should be focusing in the future. Rather than saying this drug works or doesn't work, could we do some trials before we go into this large, think of infective endocarditis. You give a generic treatment without blood culture, half the patients may be dying and half may respond, but then you send off your blood cultures and you have the appropriate drug resistance characteristics of the bug, you treat the patient with the right antibiotic, they're going to improve. We are not applying a similar strategy yet in this disease, and imaging could be a very important aspect of making that personalized decision. Spencer.
Yeah, thank you. I couldn't agree more with what Dr. DeBala stated. I think, I can really only speak to PET and amyloid PET in this aspect. And so this is, first of all, these are not approved agents yet in amyloidosis. Hopefully they will be in the future. But I think, looking back 15 years, there's a lot to learn from what was done in Alzheimer's, which is another amyloid disease. And now I think as well as tau imaging agents. And so, people were trying to measure pathology with imperfect imaging measures. And not that amyloid imaging is perfect, but it allowed the ability to see what was actually happening in the brain. And so I think that led to sort of mass improvement in how drugs were developed in that area. Eventually could be used as a surrogate. It took some time for that, but most importantly, you could see what was happening in the brain, and you could even compare drugs based on sort of shorter endpoints surrogates. And multiple PET agents are now being developed in amyloidosis, and I think Sharmila touched on this as well as Ashu's talk in particular in AL. It's very rare for the heart to be the only organ impacted, and with previous imaging, you couldn't really see what organs are impacted, and certainly with the tracers now, you see uptake in multiple organs in most patients, and so it's not. I mean, no offense to cardiologists, but it's not only about the heart, and so there are other organs to measure, and again, as Ashu stated. From the natural history, you can see that other organs actually can respond better, possibly because there are more macrophages and resident macrophages in the other organs. So I think it's important to, and potentially we have in the future the ability to measure those. My alarm's going off. I don't know if it's telling me to stop talking, but anyway, yeah. So I kind of see that as the future of imaging, being able to see what's actually happening in the pathology across the body.
Preston?
Yeah. For those of us who work with multiple stakeholders in this arena, I'm very interested in following up, Dr. Dispenzieri, on a comment that you made, a truth that you spoke, that death and hospitalization for the future is going to be an impossible standard to meet. And in this era of data sharing, if we're going to go away from a mortality survival endpoint to feels better and functions better, the experience of those of us who followed the elamiprotide example very closely, we worry about anticipation bias using registry data, because when you've got a group of people in a control group from a registry, they don't have their parents in that case, telling them, you know, you go and you walk as far as you can go because that's going to be the only way we're going to get this drug approved. Whereas the people in the registry that were the control didn't have that. And so are we to the point then where the whole trial needs to be the registry, both control and treatment, so you can get rid of the anticipation bias? And Dr. Thompson, I'd be very interested in your thoughts on that, given that you lead a division that really had interest in rigorous mortality outcomes, and the problems that you faced, I know, with anticipation bias, with feels better, functions better endpoints.
Yeah, and probably the best thing I'm going to do here is punt. I'm actually going to ask someone who's in the audience, Charu, if you want to maybe comment on this issue, because I think she really, in terms of being the lead in our cardiology space, may have some important perspectives here.
Thanks, Elisa. Preston, you raise a very important point. I think if you're thinking about endpoints that are in the field function realm or a PRO-based endpoint, I think randomization and blinding is going to be key. So if you're going to think about leveraging registry data or real-world evidence, you got to have randomization and you got to have blinding.
Prem Shravan from Pittsburgh. Matt and Ahmed, you both made a comment about comparative effectiveness, you know, of the currently available drugs. You know, the pragmatic reality that we will be able to do a comparative effectiveness trial is, I would say, really unlikely. But it made me think about the hypertension space and the heart failure space. You know, we have so many different beta blockers, calcium blockers, ACE inhibitors, ARBs, and we've learned to navigate this space without true comparative effectiveness trials. And do you think we could take a page from those disease states, and that's what will happen over time? You know, you'll ask patients once a day therapy, twice a day therapy, injectable or oral, and we'll navigate that space. Do we really need a comparative effectiveness trial?
I think you raise a very excellent point. Say my perspective, which is maybe similar to yours. I'm responding, asking the community to think about this only because this is what patients are asking us. But my own personal belief is all these therapies work and will take thousands of patients to potentially show some incremental difference. And I'm not sure that's where we should spend our energy. But the truth is that's what patients are asking. And at least in my practice, they look at me when I say, I don't know. They think, you know, who is this guy who's giving me advice? How can he not know which drug is better? And, you know, like they don't believe it. So I do think as a community, we should be responsive first and foremost to our patients. But you raise a very important point. I'm not sure that's where we should exert a lot of energy. It reminds me a little bit, you know, not to go back to cardiology days, of the Gusto trial where we studied 60,000 people to prove that TPA was a little bit better than streptokinase. But, you know, like, I don't know if that was a great use of resources and everything else. So food for thought.
I'm going to ask a question to both of you in response to Dr. Sermon's question. When you've got drugs with slightly different mechanism of action, so you've got stabilizers and silencers, it becomes a little bit more trickier than, you know, two different types of ACE inhibitors or two different types of beta blockers. How would you think about navigating that?
Actually, I was just going to say that the issue is that the drugs are distinct from each other. Even the stabilizers, if you give them to the same individual, they don't bind to the same site. So it's not the same, in my mind, it's not the same as giving, let's say, a beta blocker. You have multiple beta blockers. Some of them are selective, some of them are non-selective. And we've learned from the heart failure space that not all of them are created equal. Some of them actually had better outcomes than others. And so that's how I'm thinking about this. I agree with you, Matt, that we probably should spend our energy now on other things. But I still think that the question is valid. When you have 3 approved drugs, that every single one has a distinct binding site, distinct essentially mechanism of action, shouldn't we try to push as a community, not necessarily sponsors, but we as a community, shouldn't we try to push and understand this a little bit more? Because 5 years from now, we're going to have 7 drugs probably in total. And so are we going to then offer the patients one of the 7 options? And so that's what I think this is going to evolve eventually over time.
Justin?
Hi, everybody. Justin Groden, UT Southwestern. I want to congratulate all of you because this is great to start the day off with such an evocative conversation. So my question, and again, my comment is not meant to be perceived as nihilistic. For those of you that know me, that's not the case. I'm really posing this more as a challenge. We've talked a lot about data quality. We've talked a lot about building robust registries and data sharing. But one thing that is virtually absent or very uncommon in the literature is robust and rigorous observational comparative effectiveness. So all registries have very, very good, they've got their strengths and their weaknesses. And in particular, natural history, development of endpoints, we've heard a lot of that. But, I can tell you in the literature, a number of observational CE, they're still subject to a number of biases that are not accounted for. I think Dr. Masri had highlighted several of them. So my question really is more for the regulators in how rigorous does an observational CE need to be? Because I can tell you, at least from an editorial aspect, from journal submissions, it's really rare that a lot of those data move the bar because they might not adequately address the selection bias, the information bias, or the confounding that a beautiful randomized experiment might. And then I think we also heard about how the lack of randomization and blinding might also influence non-fatal, in particular PRO endpoints, right? I got the drug, I must walk further, or I got the drug, I must be feeling better. So you all can chew on that.
Any comments from any of our colleagues at FDA?
Yeah, and I'm maybe not understanding entirely. I mean, I think, obviously it's important to understand comparative effectiveness. And this is something very much that the community wants to understand. Patients want to understand this. Obviously, healthcare givers want to understand this. Insurers want to understand this. But I think, and maybe this has been noted already, like that is not the requirement for FDA approval, right? It's effectiveness, substantial evidence of effectiveness, and, essentially a benefit that outweighs the risk. And so I'm going to look at my colleagues and see, to what extent, I'm trying to think of, times we've gotten proposals to put something in a drug label that was related to comparative effectiveness, not coming from actually some sort of RCT. I'm struggling a little bit, but looking to others to maybe bail me out.
Well, I think what you're describing would change guidelines, not labeling. So, and I think that's what's needed, and that's why we're talking about phase 4 studies and using registries, because for a class 1A indication, you can have one randomized trial and one very strong registry-based observational study, and that will get you a class 1. So I think that there's a lot of opportunity for us to understand and change guidelines, maybe not necessarily regulation.
May I comment? It's an excellent question. And I think the heart of it is that having high quality data is not enough to do a high quality observational comparative effectiveness study. And I think the community can help build up to doing those studies with the quality that will be sufficient to move the needle for guidelines by building the knowledge base that's necessary. So, if we think of these studies, so many challenges, right? So many things that can go wrong or bias the comparison. So we need to learn about natural history, learn about data, learn about endpoints to mitigate those biases. And we have to be humble and realize some of them will never go away, right? So it's really not, we can't guarantee that we can do a high quality comparative effectiveness study for every question. But some key things to learn are prognostic factors. The better we can understand those, the better we can control bias. How do the endpoints perform? Temporal biases, how do outcomes change over time? How do they change over geography? The FDA guidance on external controls actually gives a very nice framework for thinking through all those biases. And it really takes a lot of time and research to like build up that foundation of evidence that can help you design good studies and evaluate which ones are good and which ones aren't.
Thank you. I'm going to ask a comment about, because we've been talking about registries and collecting registry data, and this is really to maybe Angela, Vaishali, even Ahmed, and I'm guilty of the same, is we all work in referral centers, and there is a big referral bias in patients who come to us. These are patients who refer to us or well enough to travel to us, and all our outcome measures are reported from big center registry data. And certainly the AL space is usually biased by these patients, and how we need to bridge the gap between maybe what proportion of patients never make it to us and what impact should we be doing, and maybe even ARC has a role here in somehow capturing the data for the patients who never make it to all of us. So any one of you.
Well, it's a good discussion. So I don't know how many comparative effectiveness study out there for AL, but by doing observational studies, in the end, there may still bias or limitations. We are developing new methods, approaches, try to minimize those limitations. For example, we could emulate a trial design. If there's an external control arm, we want to use a similar inclusion-exclusion criteria, similar endpoint definition, make them as comparable as possible. But by the end of the day, I agree that there will be realistic, there will be limitations. But still, it may still make some informative result one way or another. Yeah.
In terms of, the bias, there's clear bias. And of course, in the registries, if it's a referral center, the follow-up may not be as good. And so all of those things. I would say that it's a two-part kind of thing. Number one, yes, we don't know necessarily fully the natural history of all of our patients. And that's not a good thing. And maybe with AI and doing different types of looking through insurance and things like that, you can get a better idea of natural history of all comers. But I would then counter that the same patients that are sent to referral centers are the same patients that are going to go on trials at referral centers. So there is bias there, of course, but a little of that, there's like some correction just because they're a similar population. So again, two questions. One, what's the true natural history for all comers? But then what's the natural history of somebody who's going to go to a referral center and perhaps go on a trial? It's less bad in that second group, I would suggest.
I apologize. We're going to just, I wanted to just go back to, I guess, our two nephrologists, Eliza and Nelson, for a quick question. We're all dealing in a rare disease, but there are really rare forms of amyloid. One, an example would be ALEC2. And I fear that we're, so excited about what we're doing in AL and TTR. So I'm curious, Nelson and Eliza, if you have any thoughts or suggestions on how to move that needle in what I might call, ultra rare diseases, and how we heard regulators obviously think of rare diseases, thankfully different than common diseases. But I don't know, Nelson, you want to start? Are we leaving these people out, and how do we move forward?
Yeah, unfortunately, all the development has been with the more common amyloidosis like AL and ATTR, and actually ALEC2 is, if you look at the Mayo mass spec data, ALEC2 is now becoming increasingly more common with mass spec, and it may even overtake AA as the third most common amyloidosis. And unfortunately, we don't know a lot about it yet, and it's still one of the biggest issue is diagnostic challenge. Many of these patients have their kidney diseases attributed to other common causes like diabetes and hypertension. As nephrologists, we're not doing enough kidney biopsy on these patients, thinking that, oh, it's just diabetes, it's hypertension, and then the amyloid gets diagnosed by some other random process. And so in ALEC2, which is actually fairly common, if you look at autopsy data in Mexico City, it actually encompasses about 1 to 1.5% of the patients throughout. So in certain populations, it's actually quite common, and that I think that these registry are very important to study this, because no one center has a lot of patients, and we need to combine the data so that we can study them better. Now with the ultra rare, there are even bigger issues. You know, at the Mayo Clinic, we've been finding a new amyloid almost every year. And these are really, really ultra rare. You know, there may be 10 cases in the world, or at least recognized. And again, you know, collaboration is needed, because no center has enough cases to really make a difference. I mean, we can report on 3-4 cases of, you know, hereditary ApoE4, but does that really encompass what the real characteristic is? In fact, we're doing a study right now in ALEC2, and we were trying to come up with a predictive model. And when we collaborated with Mexico City, our model broke apart because the patients are different. And we've quickly realized that we need a lot more patients to come up with a model that actually predict, yeah, what the wider population may be. So.
Can I make a quick comment?
Yeah, sure.
Thank you. So it's a really important point about these so-called rare and ultra rare forms of disease. And Matt, I wanted to go back to your first slide where you showed how amyloidosis was felt to be rare, and now we recognize that it is underdiagnosed. And I think imaging made that transition. If we had to rely on biopsy to diagnose all these transthyretin cardiac amyloid, it would have not been the case. So having said that, I think having direct amyloid-specific imaging agents, which can image amyloid in the heart as well as in the whole body, now opens up this whole new avenue to diagnose these conditions that are rare and ultra rare, possibly maybe underdiagnosed. We will know more with the use of more amyloid-targeted imaging agents with organ imaging. So I hope that the future is going to change with amyloid targeting PET tracer imaging, where we know they bind to the amyloid.
Lisa, you had a comment, or Mitch? Sorry.
It may be keeping it on the renal side, and I know Mitch also will want to comment as well. But you know, I think speaking just in general to the kidney disease space, and certainly the rare kidney disease space, and you know, the last decade or so, we've just seen a dramatic increase in the amount of drug development in that space, and in successes as well. And I think, you know, how we got here is very much sort of what was highlighted at the beginning of this meeting. You need the village, right? You got to have the patients at the table, the basic scientists, you know, the clinical trialists, the, groups like, obviously the regulators, et cetera. You also need to have or understand what's important to patients, right? What matters to them and what their patient journey is like. And then fundamentally, you need the data, right? You need to understand and have that toolkit that's needed to sort of design what are feasible trials that can efficiently resolve questions of effectiveness. And, you know, it sounds like there are still a lot of unknowns here, but that there's tremendous opportunity to sort of bring the community together of experts in these diseases, of the patients, and to really think prospectively about how to design, you know, prospective studies, registries, where you can really map out sort of and learn about what the important prognostic factors are. And ultimately, you know, in contrast to the cardiac space where, you know, fortunately you look at these hard outcomes and feel function, the kidney space is, I would say, almost entirely surrogates, especially are rare. So it's really critical then to develop these, you know, registries, prospective studies to get the data to validate these surrogate endpoints, not just to know what could be a surrogate endpoint, but sort of how big a change you need in it for it to be meaningful. Mitch, do you want to add something?
The only thing I want to add is take it back to the question about AI and finding these patients to generate registry data, and then also finding these ultra rare patients. This is where, you know, these algorithms can interrogate a multifaceted component of an electronic health record, look at, you know, in the cardiac space, we've looked at hypertrophic cardiomyopathy using electrocardiograms only, but could integrate imaging data, biomarker data, laboratory data, natural language processing from charts, and help, you know, needs to be curated by a person still, but they are poised to be able to really help find these patients and identify them, and could, you know, then be used to bolster not only registry data, but to find patients who are in the community, not at the academic health centers. And I think that is the opportunity here, is you could build relatively inexpensively an algorithm that could then be relatively cheaply pushed out to colleagues in the community to find these patients, whether they be cardiac, multisystem, AL amyloid, or your other rare diseases, and then you could much better study them.
Thank you. We might go to the microphones. We've got quite a few people queuing up for questions.
Thank you so much, Eugene Berlassi from Columbia, New York. Great discussion so far. I just had a practical question about a data sharing that we'll talk about. It's notoriously very, very difficult to share data between institutions. Mostly the big centers carry the big data, but it's just very difficult. And others have tried with cars and all to do this kind of thing, but it's difficult. And everybody's trying to build amyloid centers, and a lot of people designate themselves as center of excellence. And there's no currently governing body to kind of designate those designations. And I wonder if ARC is in a spot to help with that. And to Isabel's point about incentivizing others to share the data, can that be implemented as a requirement to share your data with either ARC or AHA or whoever is the holder of that data as to be part of the center of excellence, so to speak?
So Isabel's right behind you, so maybe she could respond and I could ask AHA.
Yeah, I think we have to figure a pathway, and I think one of the most important things is also to recognize what are the barriers and address those. So I think there's a whole number of things that come into play. I'm going to just jump ahead quickly as I'm here, just to very quickly. I just wanted to make sure that we kept a patient-centric viewpoint. And so a couple of things that come to mind, and it is amazing that we are moving away from lives longer. And I think as we think about from, as Dr. Comanzo has said before, survival to survivorship, how important feel better is. And so as we think about developing these registries, making sure we develop and validate the tools that really understand how patients feel better. The other thing, and we've talked a lot about comparative effectiveness and the challenges and how huge those trials would be, but there were two great slides from both the presentations earlier this morning, which even to the layperson were comprehensive, about the heterogeneity. So from a patient perspective, a patient wants to know, what is the right treatment for me? And possibly as we look at subpopulations, if we could clarify that, that would be incredible. And thankfully, I've forgotten the third thing, so I will hand off.
Yeah, I would just comment, I think we also have patients who are unbelievably willing to engage, and this is a unique community. So I think we're underutilizing patient-reported outcomes personally, but
sorry, the last thing I do remember now is as we're talking about, we fail at ARC, and it's the first thing I would absolutely say in reaching a diverse community. We have over 1,000 patients participate in our study, but they tend to be educated. As we think about reaching out into the community, even if we identify the patients, we really need to look at hub and spoke. We cannot expect patients to travel to an ivory tower, even if we move them closer to them. So really, we have to think differently about how we do this and ensure we get the quality of data, but we need to have a different model to do that.
Sarah?
Yeah, great discussion. So my question is a little bit on the practicalities of some of the things that were touched on with AL therapy. So Ashu's slides were amazing, showing us that everything we're working on, and like we've got a BITE protocol for elrantamab, which is going amazingly. So just kind of thinking of that, obviously we have an effective therapy. The easiest patient population to study in AL is now the relapsed population. So if we're able to show a very clear effectiveness of a therapy in those with relapsed, is the kind of regulator labeling, would it just be for relapsed AL amyloidosis as opposed to all AL amyloidosis, specifically thinking about these novel therapies? Because then for the wider group, the temptation is to label your patient with myeloma/AL overlap, which I think has in some way tainted a lot of data that's been collected from real world, because people are labeled with the myeloma overlap to allow them get these novel therapies that we know are highly effective second, third line therapies. So that's my question.
I guess your question I could probably bounce to our regulatory colleagues, because your question was if we show something was very effective in relapsed disease, could it have a label for everything?
Yeah, and I always stay away from commenting on things that are in another division's jurisdiction. So I think I am not going to say anything. I don't know if anyone else wants to comment.
If myeloma is any judge, the answer would be no, right? I mean, but I'm the regulator.
And I think this last comment is about the payers and, myeloma/AL. I think payers are getting smarter because these therapies are millions and millions of dollars. So they are getting smarter and they are asking for prior authorization with reports of bone marrow biopsies and other criteria for myeloma. And this is from personal experience, and it has happened over and over again, which was not the case, few years ago. But these therapies are extremely expensive. So I think that it is getting more challenging.
But I think it highlights the need for trials in pure AL patients, because then if you have a trial in patients that have no myeloma and is pure AL, then the insurance can't argue with you to say, oh, they don't have 10% plasma cells.
And also, I'm going to make a comment on that. From a patient perspective, it's really challenging because they often have a very benign clone and they're labeled with a cancer label. And it really changes their perspective, and it takes a lot of explaining to the patient in terms of why do I have a cancer label when I don't actually have a truly malignant clone. And the psychological impact of just seeing that myeloma label on the letter can take an hour, 2 hours of discussion. And not just once, but many, many times, because they will suddenly forget all the discussion they had 3 years ago with you and then come back to you and say, well, actually, I've got a cancer and I'm going to die from this. Well, actually, it's the amyloid which is going to be the problem, not the plasma cell clone.
Yeah, totally. And I find that kind of is an argument I have with some of our non-AL amyloid specialist hematologists to remove that label when the patient hasn't had an organ response and I want to bring them to a solid organ transplant. And I'm like, please get rid of that myeloma label. I don't know why it's there. Yeah.
Hey, hi, everyone. I'm Leonard Chu, one of the UT Southwestern cardiology fellows. Thanks so much for having me. Amazing discussions about cost-effective or comparative effectiveness and, even combination therapies for these drugs potentially, especially in the ATTR space. My question is on cost effectiveness. I think maybe potentially some of these discussions may be moot. Why would you have two therapies if one therapy costs $250,000 per patient? My question is there an appetite for strategies or implementation trials where, you know, certain groups can implement strategies for co-payments or coverage for patients. I referenced kind of the post-MI or MI-free trial by Nitesh Chowdhury back in the day, where they offered full coverage for preventive medications for patients after MI and found kind of benefits there in terms of a cost perspective for insurance companies as well. It may not be possible in this space, but I'm wondering if there's ideas to kind of improve that type of implementation where there's a win-win-win and improve the financial toxicity for our patients.
I mean, I would comment on there's not only heterogeneity in amyloid, but in the US healthcare system, it's beyond heterogeneous. And I just can't envision that at this point for drugs that cost this much money, there's going to be blanket coverage by anybody. I think, to be honest, most of the insurers do their best to try to get patients who have amyloid off their rolls. I mean, we've seen that recently. So, you know, this is the frustrating part, somewhat heterogeneous, but that's my feedback. I don't see a short answer to the cost issue, and it'd be great if everyone had. I'm a big fan of universal coverage, but, you know, I live in leftist New York nowadays.
One quick comment about this, which is for clinical trial patients, we do have a drug like Acoramidis that patients who participate in the clinical trial have free drug for life coverage, even if their insurance covers it. This is one example. I'm not aware of any other example in cardiology or potentially even outside of cardiology where this has happened. And we should push more for this, that without the patients, the drug will never see the light essentially of approval. And that's an example where our patients who participated are actually so happy that they ended up with this. Now, there is gene editing now, for example, ongoing. That's also going to be permanent because it's in a way, one and done almost in a lot of patients. But for small oral molecules or injectables, we should use that example as a path forward to work with ARC and work with others to actually, in the rare disease space, in the ultra-rare disease space, why not? These patients helped you get there, so take care of them.
It's a great point. Bob.
Thank you. I really appreciate, we all do, I think, this great discussion. I'm Bob Gibson with Amyloidosis Support Groups. I'm also a hereditary ATTR patient. We've been, at ASG, we've been working for some time now trying to develop a natural history registry. In fact, Dr. Learn is a member of our board, as is Phaedra Johnson. Michelle Carney, who's not here, but you know, she's also on it. So we have a fairly rigorous and scientific approach at this point on it. My question is about, as you think about endpoints and the value of natural history within that, I've heard a lot of comments about biases that can be engaged in those types of things. But I always think about it in terms of, you know, what's meaningful to the patient. In all of the clinical work and the studies that have been done so far, have you come up with thresholds of what meaning is to a particular patient from their own quality of life perspective? And maybe even more generally, how might we want to think about that in terms of collecting information?
Anyone want to respond? I think this gets to the issue we were talking about before at the beginning in both of our talks, the heterogeneous population taking the average effect and not being really nuanced, if you will, in how we judge the primary endpoint and so forth. So I'm just curious, I mean, we all can learn a lot from other arenas. Is there approaches that have been used in other rare diseases where, you know, the primary endpoint is not, as we said, just live or die, but not even a primary endpoint of how much KCCQ changes, but somehow embraces the heterogeneity in the population and looks for, you know, differential effects. I see one of our FDA colleagues is standing up potentially to address this.
Thank you for that question, something we struggle with every day. So I want to say that we have accepted PROs such as KCCQ as a primary endpoint. Where we struggle is what is a clinically meaningful change, as you asked. So if we see a large treatment effect on KCCQ, I know, 10 or 15 points, we're not going to argue that this is, whether or not this is clinically meaningful. I think the places where we struggle quite a bit is when you have these small treatment effects of 2.5 and 3.7, is where we then need to look at the data further. And one of the approaches that we've been recommending, and I'm mentioning a little bit in my in DCOA and patient-focused drug development space now, but anchor-based analyses is something that we've recommended. So we ask for collection of global anchors that anchor to the specific domains of KCCQ that you think the treatment may impact. And that's one way we try to understand whether or not the observed treatment effect was truly perceived at a patient level as an improvement. And there is dilemma there because you are looking, the trial has succeeded based on a trial metric, a mean change. But then we are digging further into the data, trying to look at within patient clinically meaningful change. So one aspect of this is if you don't have a certain percentage of patients who have actually achieved a clinically meaningful improvement, it becomes really difficult to ascertain that. So, I guess the short answer is if there is a therapy that's very effective and you've had a large treatment effect on a PRO or even a functional endpoint, whether it's 6-minute walk or other assessments, we are fairly comfortable in interpreting that the therapy is truly effective. It's when you have small changes, we struggle quite a bit, and anchor-based analysis is something that we've used.
Yeah, one last comment, Nelson.
Quick comment. I
Maybe
thought
one.
Bob's comment was very insightful. I mean, we've been telling patients what is a meaningful improvement. I mean, yes, we can demonstrate that, the 6-minute walk improved, but does that matter to you? That your 6-minute walk is now 10% better? And maybe we should do a study with the patients to see what is actually meaningful improvement for the patient. I mean, we have all these tools, but are these what you want to get out of your therapy? I think that was a very insightful comment.
I think we'll now do quick ones because I can see the lights blinking red at me. Yeah.
I just have a very quick comment, and I'm going to put on my regulatory hat for a second here, and note that you can do the same anchor-based analysis that Charu just described with any functional or symptomatic endpoint. And in fact, much of the 6-minute walk test determination of what was meaningful to patients was taken from a similar perspective of saying, what was the amount of additional distance that patients said, oh, I feel better, and this is the amount that was seen. So, and in general, right, that concept can carry through whatever your symptomatic or functional endpoint you want it to be to make sure that it is important to patients.
I think we have one of our regulatory colleagues wanting to comment on this. And then we can come back to the question.
Yeah, just to close the loop there. We don't have to do this in a trial setting. You could come up with a study that is non-interventional just to try and understand how much of a change in KCCQ or 6-minute walk or other functional or peak VO2 is really perceived by patients as a change. The amyloid population is quite heterogeneous. So yeah, you're gonna have to think about how large a study you need to just evaluate symptom and function and global anchors. It's really qualitative work. So if you're going to invest a lot of time and effort, maybe registry platform is can be used in such a way to try to define what is the range of a meaningful change for these various metrics. And then you don't have to sort of redo the work in a trial setting.
Thank you. That's really helpful. Yeah. Question, last question.
Hi, Glenn Krug from Regeneron Pharmaceuticals. I'm new to this group, and I'm trying to understand. There was talk on ATTR amyloidosis, AL amyloidosis. There were a lot of discussions about things about registries, imaging, PROs. Do all of these topics apply equally to the different types of amyloidosis? I tend to think of these as very different kinds of physicians and patients. How does this group view amyloidosis? Is it a group of different diseases? Is it one umbrella where all of these topics count equally?
I think I'll probably respond to that. That's a really important and really difficult one to answer, because it started with small groups of physicians interested in amyloid who would see every single type of amyloid, and we still do that. And then the treatments have diverged in terms of treatments for TTR and treatments of AL. And even as physicians, we are diverging. I don't see TTR patients any longer. I just see AL. I used to see everyone beforehand. But then you've got these ultra-rare amyloid types, which we still see as amyloid physicians, who nobody seems to know what to do and how to diagnose and how to manage. And the imaging is very similar. The end organ dysfunction is very similar in all types of amyloid. The organ involvement tends to cause very similar types of symptoms. So there is a lot of overlap between these diseases. So I think we need to think of it as a group of conditions which cause similar problems, but clearly understanding that there is a divergence in what's happening with certainly the two big amyloid types, AL and TTR, and then all the rare amyloid types, which we still need to field as a group rather than separate them out into different buckets. And I think there may be some things which we can do together as a registry with big centers which captures outcome measures in AL and ATTR for cardiac amyloid, and the analysis is done differently. So you don't have to do 5 studies, you can do it under 1 study. And maybe that will be something that we need to think of as a community, how we go forward, not now, but in the next 5 years.
Well, thank you all. It was a very robust discussion. We're going to reconvene at 10:40, so 14 minutes from now.
All right, we're going to go ahead and get started in our second session, which is focused on the work from our working group that's focused on imaging standardization. So this session will be moderated by Dr. Ahmad Masri from OHSU and Sharmila Darbala from Brigham and Women's Hospital, Harvard Medical School, our 2 working group co-chairs. Before I hand it over to Dr. Prem Saman to kick us off, just a reminder to the folks that are on the panel, for those who are joining us virtually, please remember to just state who you are when you start talking so that the virtual speakers or the virtual attendees can follow along. And then 2, just leaning into the mics as much as possible, and for those at the podium so that everyone in the room can hear as well as those online. All right, so with that, I will turn this over to Dr. Prem Saman from University of Pittsburgh Medical Center, who will provide an overview on the imaging modalities that are used for cardiac amyloidosis.
Thank you, Kirsten, and thank you for introducing me so that I don't have to do it myself. Good morning, everybody. Thanks so much for the invitation. So this slide is probably superfluous for this audience, but I always start with that, and I will do so today, to remind us that imaging has been truly transformative for the field, definitely of ATTR amyloidosis, to some extent for AL. And by allowing us the ability to make a non-biopsy diagnosis, it has actually unmasked a previously unrecognized community prevalence of disease that has made all the remarkable developments in therapeutics much more meaningful. And of course, it has catalyzed the recruitment of patients into therapeutic trials by obviating the need for a biopsy. Imagine if every patient recruited into a clinical trial needed a biopsy. That being said, despite all this development, we have not been able to find imaging endpoints to use in clinical trials. And I would say that this is a reflection of some fundamental knowledge gaps in our understanding of this disease state. For example, we have no idea what happens in the myocardium when we treat TTR amyloidosis. Is the patient improvement related to the reduction of toxic prefibrillar oligomers, as we know happens in AL, or is it because of the natural scavenging mechanisms that remove the amyloid? Second, there's a very complex relationship between cardiac structure and patient symptoms and outcome. For example, if you showed me an echo, it would be very difficult for me to predict by looking at that echo how that patient's feeling, what their NYHA class is. And as was alluded to earlier today, we have a number of imaging metrics that reflect the effects of infiltration, but very few imaging metrics that directly reflect the amyloid burden. And as we've seen repeatedly, especially in TTR amyloidosis, diagnostic efficacy of a modality may not necessarily parallel its prognostic utility. Now, pyrophosphate scintigraphy or bisphosphonate scintigraphy has truly been transformative for the field. And I would submit here that the beauty of this technique lies in its simplicity. It's a rest-only study. No patient preparation is required. Almost anybody with a SPECT scan can do this. And it is so widely applicable, irrespective of body habitus, atrial fibrillation, renal failure, cardiac devices, and so on and so forth. So literally anybody you refer for a PYP scan or an HMDP or DPD scan can actually get it. And it is associated with a relatively low radiation dose. But again, despite the fact that we have used this now in thousands of patients, there are many fundamental knowledge gaps, starting with the simple question of what is the real-world sensitivity of scintigraphy? Now, we saw from Julian Gilmore's paper, they reported an incredible sensitivity and specificity, numbers that we rarely see in clinical imaging. But remember that in this highly selected population, the prevalence was 70%. And as Matt and Rick told us in this very insightful editorial titled Caveat Emptor, meaning buyer beware, that if you do this test in the community, the sensitivity and specificity may be different. But I would submit, on the other hand, it is not 70%. And this is a number that is increasingly appearing in the literature and in presentations. And this seems to come from this table in Julian's paper that refers not to the sensitivity of the scintigram, but to the sensitivity of the non-biopsy criteria. And the criteria includes that the serum and urine studies are completely normal. So every patient with TTR and MGUS, every patient with TTR and renal failure would be misclassified. And that would have happened irrespective of the diagnostic modality used. In fact, if you properly classify the 88 patients in that study with TTR amyloidosis and MGUS, now the sensitivity goes to 90%. And even this analysis excludes patients with grade 1. So the sensitivity and specificity are not 99% and 100%, but it's not 70% either. The other important question is, what exactly are we imaging with PYP? Here is a patient, biopsy-proven variant TTR, with sort of patchy uptake of PYP. You can see that we had to do a dual isotope study and contour the myocardium to be absolutely sure that the tracer uptake was in the myocardium. But look at the MRI in this study. It was one of the most abnormal MRIs I've seen in patients with TTR amyloidosis. So very patchy uptake of PYP, very abnormal MRI. And what is the reason for this discrepancy? And in fact, if you look at the perfusion scan done in this patient, there's very nice uptake of a technetium MIB here, telling us that there's no myocardial infarction that precluded the uptake of PYP. And quite likely, there's not a significant amount of macroscopic or replacement fibrosis. So we really have no idea what the PYP is binding to. Now, Sharmila and her colleagues showed us that if you take patients and follow them up with serial quantitative pyrophosphate imaging, you can show a reduction in the quantitative metrics on serial studies, but that has no correlation with a change in structure or function, including the global longitudinal strain. So is the reduction in PYP due to a reduction in amyloid, or it's simply because of a change in the molecular structure of the fibril with therapy? We have absolutely no idea. On the other hand, we do know that the PET tracers do bind to amyloid. So the thioflavin analogs, which include the F18 tracers, and by the way, Congo Red is also a thioflavin analog, and all of these agents bind to the cross-beta-pleated secondary structure of amyloid. And remember that all amyloid, irrespective of the protein of origin, has this common structure. And that's why these agents are pan-amyloid agents. Also, evusemetide binds to the negatively charged glycosaminoglycans that exist both in the fibrillar and non-fibrillar components of amyloid, and is also a pan-amyloid agent. We have very, very encouraging data of highly sensitive detection of small amounts of amyloid with both of these agents. The top panel there is an image from one of Sharmila's presentations, showing that even in patients with AL, treated AL, who are assumed to have a very good clinical response, you can show small amounts of residual amyloid by sensitive imaging with PET. Now, on the bottom panel are examples from evusemetide imaging showing no uptake in healthy controls, good uptake in patients with clinical ATTR amyloidosis, and also uptake in patients with suspected amyloid who were otherwise imaging negative. So very good evidence of sensitive uptake of tracer. And on that other slide, on the other image there that was given to me by Ahmed, showing that with evusemetide, you can actually very nicely detect extracardiac amyloid, which is a particular advantage perhaps in AL amyloidosis. Now, evusemetide seems to work slightly better in TTR compared to AL, and the F18 slightly better in AL compared to TTR. So we now have bisphosphonate scintigraphy and PET, and PET is very good for early detection of disease. But I would submit to you that for many years, maybe for a decade, we are not going to be in a position to screen thousands and thousands of patients with PET. We simply don't have the ability to do that. And on the other hand, we have SPECT that is widely available, but questions about how early can we detect disease. If you look at that SPECT scan in that image, what would you call that? Would you call it negative, or would you call it grade 1 amyloid? In fact, I would submit that even the more fundamental question of how early should you treat TTR amyloidosis, should we treat asymptomatic patients, that is a question that as yet remains unsolved. Multiparametric cardiac MRI provides a wealth of information, including many morphological abnormalities, characteristic subendocardial enhancement patterns, and very importantly, the ECV, which is a unique reflection of what's happening in the extracellular space, may be specifically suited to evaluate patients with TTR amyloidosis and AL amyloidosis. In addition, another reflection of the expansion of the extracellular space are these abnormal gadolinium kinetics with the myocardium nulling before the blood in the teen version SCOUT images. Now, different labs use MRI differently. In our center, almost everybody gets an MRI. But where we find MRI the most useful is at the extreme ends of the spectrum of abnormality. So if you have an equivocal PYP, for example, a completely normal MRI is very reassuring. And on the other hand, if you have all the abnormalities, especially if the ECV approximates an arbitrary number of 50 or above, that is amyloid unless proven otherwise. If the patient has not had a transmural myocardial infarction, there's very few other things that bring the ECV up to that number. So this is sort of how we use MRI in our lab, but I know there's a lot of heterogeneity in how this is used. Echo, of course, is usually where the question of amyloidosis first is raised. And this combination of a thick ventricle with a small pericardial effusion, as you can see here, is very, very suggestive of an infiltrative cardiomyopathy. There are a number of other abnormalities. Almost every structure in the heart is affected with amyloid. Important to note that asymmetrical septal hypertrophy can occur. And it's important that these large atria that are not only large, but they're mechanically stunned, makes the risk of thromboembolic phenomena in patients with ATTR and AFib very high, and a risk that is not adequately captured by standard risk scores such as the CHAD score. The focus, I want to talk a bit about the fallacy of the ejection fraction. So when you get an echo report in patients with amyloid and it says that the EF is 60%, this is a false sense of security. As you can see in that image, the moving image, a very thick ventricle, a very small cavity, ejection fraction of 60%, the stroke volume is still very, very low. And these patients are very dependent on the heart rate for their cardiac output, and this is why they feel so much better when the beta blocker is stopped. Now, it turns out that the GLS, the global longitudinal strain, is the most sensitive detector of systolic dysfunction. And this is because the GLS reflects the longitudinal fibers in the subendocardium, in the subendocardial space of the myocardium. And as you know, most pathologies start in the subendocardium and then progress transmurally. And the radial fibers that are responsible for the EF are situated in the mid to the epicardial regions. So the GLS is very sensitive, but it's important to remember that the GLS is very much affected by loading conditions, and it may be a marker more of the effects of infiltration, certainly does not distinguish ATTR from AL, and it may be affected by a number of other things than the amyloid burden. Now, the focus, of course, in contemporary imaging is on our ability to do serial studies in ATTR and AL. Now, I'm showing you a few studies here. On the left side is a recent study from Mariana Fontana looking at about 100 patients who were treated with patisiran and comparing them to patients who were not treated. And what they showed is a small, single-digit percentage reduction in the ECV. And the ECV was derived by sampling just the septum, okay? And they showed that in the untreated patients, there was no such regression and there was progression. Now, on the right side are serial studies with evusematide, and a very interesting observation here that I think Sharmila alluded to earlier. You see no change in the cardiac uptake in this patient with treated AL, but you can see very good organ response in the liver, spleen, and in the other organs. And this particular prowess of this agent for extracardiac imaging may also be partly related to the iodine tracer. And this is something, the iodine tag, which is something to keep in mind. And here are some data, the recent data from HELIOS-B, showing again that there was a decline in the rate of deterioration of GLS and stroke volume, very small changes. And none of these therapies actually brought these metrics back to the baseline, showing that there was no reversal of disease, only an attrition in the rate of decline. Now, the unifying characteristics of all these studies are the following. The changes in measured parameters were really small. And the measurements were made in very highly expert core labs or in single center studies. And the interpretation of these changes was made in comparison with an untreated or placebo group. Now, absent these conditions, I'm not sure that we can translate these changes to individual patients in the community. And it is striking that there's a frequent lack of concordance between changes in the amyloid burden and structure and function, the cardiac structure and function. So, what is the current lay of the amyloid imaging land? If you look at the different modalities, bisphosphonate scintigraphy today is the only technique that can reliably, in most patients, distinguish between ATTR and AL disease. This is today the only method of a non-biopsy diagnosis of TTR amyloidosis, of course, when it's performed in conjunction with serum studies to exclude biochemical studies to exclude a plasma cell dyscrasia. Now, what are the metrics that specifically measure amyloid versus the disease effects? And today, the only approach is PET tracers that we know that definitely bind specifically to amyloid. And for disease monitoring, there's a little bit of data with CMR, and hopefully we'll have more data with the specific PET tracers. So if you were to ask me for a wish list of the ideal cardiac imaging technique for amyloidosis, I would say that for screening, you need something that's widely available, widely applicable, low cost, high accuracy, low radiation. And you know what? We have it with bisphosphonate scintigraphy. For disease monitoring, we need very sensitive detection. We need a quantifiable measure of amyloid burden to monitor disease activity. And the two candidates now, contemporary, are ECV, that seems to do this. And then, of course, we're watching the space with the PET tracers. Thank you very much.
All right, thank you very much. Dr. Soman will proceed with the next talk by Dr. Jeremy Slivnik. He's going to speak about use of AI and imaging technologies.
Hi, everyone. Thank you very much for the opportunity to speak today. We're going to talk about the use of AI in cardiac imaging and for cardiac amyloidosis, and specifically focus on echocardiography. I think it would be challenging to cover all of these within a single talk. However, I think most of what's going on in echo is also going on in these other imaging techniques, and hopefully this will serve as a template to understand what's going on in the AI space for cardiac amyloidosis. We'll also, in addition to discussing the potential use cases, we'll highlight some of the pitfalls and future direction in AI in the field. So as a whole, I think we've seen over the past few years that AI has shown really transformative potential to improve many aspects of our day-to-day life. And this is no different for medical imaging. We've seen uses and utilization of AI across the spectrum of cardiac imaging techniques, ranging from helping us to acquire images, view classification, automated measurements, even automated reporting based on these measurements, and up to even disease diagnosis. And this has been shown throughout cardiovascular imaging, but I think we've seen that this has really made its way into the cardiac amyloidosis field, particularly within the past few years. So I think before we talk about where AI has come into the field of cardiac amyloid, I think it's helpful to understand what is really needed and what are the current opportunities in this space. And I think, as Prem alluded to, we really need expanded access to cardiac imaging testing, and we need to improve the imaging-based screening, particularly in patients with earlier disease. And in addition to this, there's an increasing burden of studies on readers in cardiac imaging. And so we need to find ways to improve the efficiency of image interpretation and screening so that we can help to increase the throughput of patients, particularly as increased recognition drives increased referrals for screening for cardiac amyloid. And lastly, from a clinical trial perspective and a treatment perspective, we need reliable tools that can help us to reproducibly and accurately monitor patients in response to therapies. So let's talk a little bit about how AI can potentially help for disease detection. I think as we heard about, impairment in global longitudinal strain is really a hallmark feature of cardiac amyloid, particularly this apical sparing pattern. And this is really something that we're using day to day to help screen patients for cardiac amyloid, as well as increase relative wall thickness. However, there's increasing recognition that these markers, while previously shown to be accurate, may lack the sensitivity and specificity to function as standalone screening tools. We can see here in this study that the sensitivity and specificity was 72% and 66% respectively, highlighting the need for more refined approaches to really get the most yield out of echocardiography. And I think as we've sort of heard looking at these imaging techniques, there are many advantages to tools like nuclear imaging and CMR, but I think we recognize that echo is often the frontline tool that these patients are being screened with. And so patients that are getting missed by these conventional parameters may fail to ever get referred for these imaging tests. More recently, there's been the development of multi-parametric tools that have helped to overcome some of these limitations by utilizing either multiple echo parameters in the case of the IWT score out of the NAC, or in the case of the TCAS score, utilizing a combination of echo parameters and clinical variables to help refine the accuracy. And these have shown promising potential. But, I think of these things from the snapshot of the echocardiographer. And, I think when I'm on the 50th echo of the day, it's really challenging to employ multi-parametric models. I think it's hard to scale this. I think these can be done in clinic, but it's very challenging from our standpoint. So, I think this has sort of created a potential role for artificial intelligence. And I think it's helpful before we dive into some of the data to just get a little bit of an understanding of what the types of AI tools are and how they could potentially be applied. So, we have sort of, most of the models that we'll be talking about will be deep learning models. And so these are basically, it's a subset of machine learning where the data is basically input into a convolutional neural network. And there is no feature extraction, meaning that the deep learning network learns from the images or for whatever the data provided to it and kind of creates its own unique insights in order to predict what the ultimate ground truth is. Whereas machine learning, there's first underlying feature extraction where humans or other sources of data are basically curated into parameters that are then input into the model to ultimately yield a prediction. So this was one study that we did looking at a deep learning model for the detection of cardiac amyloidosis. This was a multi-center collaborative between 18 sites as well as the Mayo Clinic, in which we developed a model which input apical four-chamber views to differentiate cardiac amyloidosis from phenotypic controls as defined by the gold standard of either invasive tissue biopsy or non-invasive diagnosis with nuclear imaging. And we found that the accuracy was high for this, with sensitivity of 85% and specificity of 93%. And based on this ultimately led to FDA certification of this technology. We also found that the AI model outperformed the multi-parametric risk scores in a subset of patients that had this echocardiographic data available to them. Similarly, we worked with another company to develop a similar algorithm utilizing apical four-chamber images to differentiate amyloid from phenotypic controls that yielded similar accuracy and demonstrated similar performance to multi-parametric scores across both AL and ATTR subsets, again leading to FDA certification. And so based on this, we now have two FDA approved technologies that can be utilized to potentially screen patients in the real world for cardiac amyloid from an echocardiogram. Now, this is, I think, really interesting data because this was, largely we utilized registry data of patients with confirmed cardiac amyloid, but this doesn't really answer the question of whether AI could potentially be employed for early diagnosis. And Dr. Kara and colleagues at Yale, they've developed similarly deep learning models for amyloid detection, and they actually looked back at ECGs and echocardiograms years prior to a diagnosis of cardiac amyloidosis to see how these models could perform. And they compared patients that were ultimately diagnosed with control patients that had suspicion but were ultimately ruled out. And I think one of the really fascinating things that you can see here and really encouraging is that, we could see that the signal, there starts to be divergence in the AI probability thresholds years prior to their clinical diagnosis of amyloid. Even up to, up to 3 to 5 years, we're already seeing some divergence. And I think this really highlights the potential for early diagnosis. But I think really we're talking about earlier, because I think we've always noted that we're, even at the time of diagnosis, there were probably many missed opportunities. And so, I think we can say earlier diagnosis may be possible, but I don't know that this necessarily represents earlier in the true sense. So I think despite the many potential promises in this area, I do think we need to acknowledge several potential unanswered questions. For one, we don't yet know the real world performance of these models and how they do when they're released into the wild, particularly in situations where we've already identified many amyloid patients. And so we're screening those patients that have yet to be diagnosed. Is that going to result in more false positive tests? And additionally, we don't necessarily know the correct utilization. How do we enrich our pretest probability to appropriately make sure that we're screening high enough risk patients that these models are going to function in clinical practice? We don't yet know whether AI can truly facilitate early diagnosis. We think it can potentially promote earlier diagnosis, but we don't know yet about early. And basically how this can be expanded to clinical practice. We don't yet know how AI tools can be integrated with established clinical risk markers, right? We think of clinical decision making as being an integration of many parameters, including red flags, clinical demographics, and the imaging parameters are part of that. But we don't really know how to integrate all that data into clinical decision making yet. And therefore, it's hard to know the exact optimal workflow. And I think another factor is the reimbursement model. I mean, I think we're seeing multiple potential pathways for how these can be billed for. And I think that's something that really is going to have to be learned as these models make their way into clinical practice. And I do think we need to discuss the potential challenges as well. I think with black box AI, there's always a lack of explainability, and this can both, hinder clinician and sonographer trust in AI algorithms, as well as can, make it difficult to understand the insights that it's coming to. Data sharing and governance are going to be critical as we deploy these. And there are certainly to be unforeseen financial and administrative burdens as we screen new populations, identify new potential patients. This is going to result in a lot of testing. And what we all want, which is potentially more patients with amyloid that are identified that are then going to need to be started on therapies. And I think this is going to be something that's going to be challenging to navigate. And lastly, there are the legal and ethical ramifications of AI misclassification. So another area I want to touch on is measurement accuracy, reproducibility, and efficiency. And I think this is an area where AI is already helping and could potentially really be beneficial in amyloid. I think, we've talked about the issues with inter-observer variability and how that can affect our ability to monitor patients with imaging in response to treatment. And this is data from the HCM space. And as of yet, we really have a paucity of data in amyloid. But here we can see that this was a study looking at measurements of wall thickness, which is really critical to how we monitor amyloid patients. And we can see that there's massive variability in experts, and that with the use of AI, we could potentially reduce that variability without compromising accuracy. So I think this is definitely an area of potential promise, although we still need to explore this in the amyloid space. This was a study looking, as we know, the importance of global longitudinal strain, and similarly showed that AI algorithms for automated strain measurement resulted in less variability compared to expert readers. Again, highlighting the potential for the two most important parameters that we utilize in clinical practice. So what are some future directions for AI-guided measurements? Well, I think for one, we need to understand whether AI can similarly improve the accuracy and reproducibility of measurements in amyloid as it's been shown in the general population, as well as in the HCM space. I mean, I think there's definitely reason for optimism, but I think we do need to show this specifically in our patient population. And if so, what is the potential of those improvements on our ability to diagnose patients, monitor patients in response to treatment in clinical practice, as well as the utilization of imaging-based biomarkers in clinical trials? Lastly, I want to touch on patient access to imaging. As many of you may or may not be aware, we're really entering a workforce crisis in echocardiography and imaging as a whole. We have an increasing demand on readers and sonographers with numbers of studies done per year, but we're increasingly having less and less trainees being released into the workforce. And this has created a burden on and a potential barrier on our ability to get patients successfully diagnosed and screened. So one potential technology that I thought is really interesting is the concept of using AI-guided point-of-care ultrasound. So these are technologies that can basically almost use like a flight coordinate system to help guide novice users through the acquisition of an echocardiogram. And this offers one potential solution to the scalability issues of having to do increased exams. And in this study, we found that novices demonstrated similar ability to obtain diagnostic quality images as compared to certified sonographers. And this was, I thought, a really fascinating study, again, from Dr. Kara's group, where they showed that not only could they acquire these POCUS images, but that these POCUS images could actually be applied to artificial intelligence algorithms for amyloid detection with reasonable results. So showing the potential that these images can not only be obtained, but could potentially be leveraged for our purposes of screening patients for amyloidosis. So in conclusion, I think AI can bring a lot of potential value to the field of medical imaging, both through the improvement of allowing image acquisition, automated measurements, and automated reporting, as well as the potential to diagnose disease through deep learning. And through that, I do think that this is going to help us to augment the capacity of imaging to help in key strategic initiatives for cardiac amyloidosis care, including improving the potential for timely diagnosis of cardiac amyloid, improving our ability to monitor treatments in clinical practice, and potentially improving it as better biomarkers in clinical trials. Thank you very much.
Great, thank you. Next, we have Dr. Rick Ruberg, who's going to talk to us about the ECHO study overview.
Thank you very much. First of all, I want to thank ARC, Isabel, Kristen, and Jamie for inviting me and letting me speak. And on behalf of Ahmed and Sharmila, I'm going to be presenting basically the study that we have arrived upon from our working group. Okay, so what are the goals of the imaging working group? We have very defined goals. The purpose of this, our working group, which is at this table up here, is basically to devise imaging biomarkers and establish the reproducibility of these markers for disease progression and prevention, and prognostication. And I think one of the things that I observed upon from the previous talks, Prem did an amazing, elegant job providing the overview of imaging for amyloidosis. And Jeremy, if you were not convinced that ECHO was where we should start, you weren't paying attention. That was a beautiful presentation about AI. So over the next 10 minutes or so, we're going to talk about how we can integrate some of the things Jeremy talked about into what we propose to do. We propose to develop study protocols of sufficient rigor to basically establish the capacity of imaging markers that could be used as disease markers. And I wish I was able to add a few slides from this morning's presentation, but I want to just reflect on imaging in general and go off script for a few seconds. First of all, I totally agree with Sharmila that imaging is critical because it allows us to see exactly what's happening in the heart. Sure, I mean, endpoints such as survival, hospitalization, PROs, and even downstream endpoints are obviously critical for registration purposes, but understanding the efficacy of a treatment is what imaging can do, number one. Number two, imaging biomarkers are not like blood biomarkers. Blood biomarkers have a narrow coefficient of variation. When you order a proBNP and a creatinine or a troponin I, that measurement is going to be fairly precise in respect to the total value of that number. Imaging is different. There's a considerable amount of subjectivity in imaging measurements, which can be overcome in many ways by automated interpretation, which we'll talk about. But I think that's a critical piece. And if we're going to base assessments of therapy or even registration, which we probably wouldn't, on an imaging marker, we have to understand the inherent subjectivity. And the other point that it was made this morning that I wanted to echo that Matt made and Ashu made was that there's considerable heterogeneity of response. And so when we look at the mean change in, say, global strain in a population, some of those patients will have gotten better, some of those patients will have gotten worse, and you're not going to capture that variation in response by looking at the mean number. And the final question, which we're not addressing, is a clinically meaningful difference. We're going to talk about a measurable difference and a reproducible difference, but the clinical implication of that is to be determined by other studies. So our goal, as I just basically said, was not to determine which markers, but how can we determine the reproducibility of these markers. And so as Jeremy mentioned, we've chosen echocardiography because of its global availability, its absolute safety, and it's relatively low expense, and it is the gatekeeper for every cardiac heart failure imaging, any study. There are really no contraindications to echocardiography, unlike MRI, and not so much for nuclear imaging. Acquisitions are short. They can be done at the point of care. We've talked about automated interpretation and acquisition tools, and the capacity, which Jeremy also alluded to, miniaturization and handheld devices. So echo is a very democratic imaging modality. It can be done anywhere in the world by essentially anyone, even people with minimal training, unlike the other imaging modalities, which is why we've arrived at echo. So what are the measures that we can look at by echocardiography? Echo provides indices of structure and function. The structural measurements you can see here, wall thickness, the chamber, the diastolic dimension, volumes, and then derived things like relative wall thickness, and we can measure volumes of all cardiac chambers. Structure is very important. Many studies that I've led and others, many people in this room have led, have had structural criteria that patients have to meet with a minimal wall thickness, for example, or functionally a minimal ejection fraction or a maximal wall thickness, whatever. These are things that need to be, that are part of every echo study. So functionally, we look at ejection fraction, we look at right ventricular function, that's what TAPSE stands for, tricuspid annular. Planar systolic excursion, don't say that too many times quickly. Global longitudinal strain ratios, stroke volume ratios such as the contraction fraction, etc. And we can also assess diastolic function. So echo provides, unlike MRI and nuclear imaging, a really complete assessment of the function and the structure of the heart. Again, another reason why it's an attractive modality to first approach. Now, I'm going to talk a little bit more about strain. This is already introduced by both Prem and Jeremy. But the echo world has coalesced around global longitudinal strain. And so the concept in global longitudinal strain is you're looking at the longitudinal deformation of the heart in the long axis, and you average it across all the cardiac segments. Other strain variables have been reported and are currently reported, but the American Society of Echocardiography and the European Association of Cardiovascular Imaging has really coalesced around longitudinal strain, and it has become essentially the standard of care in many cardio-oncologic imaging practices. Prem already alluded to the apical sparing pattern, which has been described in amyloidosis, and there was considerable enthusiasm about it at its initial description as a useful diagnostic tool, but turns out to be helpful, but not. But other disease states can also produce a similar pattern. And so we've basically kind of like adapted global strain generally as the ratio. So the advantages of global strain that use speckle tracking, I'll talk about that in a minute, it are that essentially it's angle independent, and it's relatively easy to apply and interpret, and it has relatively high reproducibility compared to older strain techniques which use Doppler imaging. Now, the way strain actually works is the ecosystem is looking at each individual speckle or echocardiographic reflection and looking at how those reflections move towards and away from each other, integrated over time. So it takes the displacement of each of those individual speckles, integrates it over time, and creates strain. But the strain measurements are very subjective to where the contours are actually drawn and the measurements are taken, and that introduces error. So the ASE and the EACVI began to explore the variability of these measurements, not in amyloidosis, which is why we're talking about this study today, but in non-amyloidosis patients. They took 63 not healthy volunteers. These are patients who had cardiovascular disease, typically scar and infarction, and looked at their echo strain reproducibility. Now, this figure illustrates the various different vendors there. Most vendors, most of us, I would say, in the amyloidosis specialty centers use either a GE, a Philips, or a Siemens platform. But there are variation in the ways in which the strain calculations are performed. And you can see on the right side of the screen the differences, the absolute differences, around 4%, 3%. Now, that's a fairly sizable difference when you're talking about a number where the variable for normal is around 17 or 18%. So 3% is a fairly high coefficient of variation when you're looking at a number of that size. Again, in contrast to, say, a proBNP or a troponin. The other kind of nuance here is that there has been some discordance about when to take the strain measure. But again, the American Society of Echo has coalesced around end systolic strain, which is the strain which you can see there at the end of systolic ejection, as illustrated by that Doppler waveform. Now, automated interpretation, as Jeremy introduced, has tremendous potential. I want to call your attention to this recent publication about a month or two ago from the Cedars-Sinai group, David O. Yang, looking at an open source protocol called EchoNet. And they basically looked at sonographer annotated echoes versus the EchoNet analysis. And what they did was, instead of doing an automated interpretation, an artificial intelligence classification of amyloid yes, no, which is what Jeremy showed in his European Heart paper, this is a technique to actually quantify wall thickness, ejection fraction, not ejection fraction in this study, wall thickness, diastolic dimensions, and Doppler measurements. And the study showed very good and reproducibility of the automated interpretation versus the human sonographer annotation. So what we propose to do is to leverage such techniques into an echo reproducibility study. So, and now for the next few slides, I'm going to present what we propose to do, and it will be obviously open to discussion at the end. So the study is led by Sharmila, Ahmed, and me, and Jeremy is leading the core lab interpretation for this study. And we are trying to be as broad as possible here. So any patient with AL or ATTR amyloidosis, and essentially, we're not proposing any exclusion criteria. You could exclude for atrial fibrillation, but we don't want to do that. We want to make it real world. You can exclude for patients with paced rhythms, and we can talk about that. But again, we want to make this as broad and broad and applicable as possible to most patients. So we propose to basically take all comers who consent to be in the study. What we propose to do is to recruit from 3 sites, Brigham and Women's, Sharmila's site, OHSU, which is Ahmed's site, and BMC, which is my site. And then we would then send the images to University of Chicago, where Jeremy will perform the core lab analysis. And we're proposing a relatively small sample size of 200 patients, equally distributed between AL and ATTR. And what we propose to do is essentially a reproducibility study with a standard clinical echocardiogram. Clinical echocardiography is standard of care for all patients who have AL-ATTR at our institutions. So they would acquire a clinical echocardiogram first, and then they would have a second echocardiogram performed that we then use to compare. And we would propose that in the second echocardiogram, we would measure standard structure and functional measurements and global strain. So potentially a bit more limited than a complete echocardiogram, so the patient doesn't have to, basically endure all the measurements that we would do in a complete echo. And the second echocardiogram would be performed by a different sonographer. That's another important point to control for variation in sonographer skill. And so in the analysis, we propose to have the echo analyzed by two different humans, as well as an automated interpretation algorithm using the US2 AI platform that Jeremy had presented some data from earlier. So we propose to look at inter-study agreement, so test and retest. How well do these two echo studies, basically in the same person, essentially at the same time, right after the other, are they reproducible? This is important so we can understand in amyloid patients, the reproducibility of these measurements and the capacity of them to basically be used either to follow response to therapy or determine eligibility for studies across and broadly implemented studies. So we propose to look at the reproducibility between the two human interpreters, as well as the mean human interpreter result and the artificial intelligence result. And we would look primarily at the linear measurements, and we would also look at the global strain. And we would use the vendor agnostic TomTech auto strain analysis platform, which uses DICOM images only and doesn't require a specific data preprocessing or storage as the other vendors may require. Statistically, we would look for interclass correlation coefficients between the two measurements. We'd look at Bland-Altman measurements to look at the range of measurements for bias with one technique or another, one analysis or another, and determine the coefficient of variation in amyloid patients, as has been kind of broadly done in non-amyloid patients. And we also would importantly stratify by ecosystem, so we want to understand if imaging with one ecosystem or another provided bias. This is my last slide. Just in terms of cost, we propose that the first echo would be performed clinically and the second one would be billed to the study. We arrived at a reasonable echo charge at our institutions, and also the cost of coordinators. We also proposed to a fee for the core lab interpretation and an indirect cost. So the total cost of around $200,000. Not quite a rounding error to some of our partners in the audience, but still not a huge amount of money that one could put together. So we think this is a reasonable investment. I'm here now looking for money. We think this is a reasonable investment because we do think these will provide critical information that will then be utilized by the other working groups as they vet these variables for their clinical utility. So with that, I will end my presentation. Thank you all.
Well, thank you all for these great talks. Just a reminder that when you speak, please introduce yourself so that the online audience can know who's actually speaking. So I want to ask Prem first, follow up on your presentation. So for scintigraphy, one issue is that it's a confirmatory test. So most patients, by the time they get to a scintigraphy test, they've already had high suspicion of having the disease. And one of the issues I think that imaging needs to adapt to is how can we actually start picking up early disease? And I would ask you that I don't think that actually scintigraphy has been challenged with that concept, that is the non-heart failure early disease, because we always use it as a confirmatory test to replace biopsy, correct?
That's a great point. Although, for example, in the variant disease population, we scan patients, we scan relatives of an index patient who carry the gene and have no symptoms. I think I want to recount a conversation I had with a PCP in the corridor. And he said, Prem, the most important thing establishing the amyloid center has done is that it's made us all think of amyloid. And if you think of what has happened in the past decade, it's quite remarkable. And I don't think you can dumb it down so much that this whole process can be automated. At some point, you have to have a physician who thinks about this, refers a patient, and we know that you can have a positive PYP when the echo looks normal. So there isn't a sort of standard approach that you can take. It's dependent on the patient. It's dependent on how strongly you suspect amyloid. If you have somebody with a very high suspicion of amyloid and the PYP is negative, we still biopsy those patients. Whereas if the clinical suspicion is not that high and the PYP is negative, you stop there. And so I agree with you. I think, what is the best method to detect this earlier? And I think that's a human being that thinks about the disease, and then we follow up on that.
Yeah, so I just, Sharmila Durbala here, just wanted to extend on Dr. Prem Soman's comment. I think the next phase of developments are earlier and earlier disease detection. The advanced disease detection, we are doing quite well. The early disease detection is where we need more advances. There are some studies, so clinically, we are looking at patients who are symptomatic presenting with suspected amyloid, but there are epidemiological studies from Mayo Clinic, Omar Abuazzetin, SCAN-MP, and we are looking at some of the cohorts as well, where you are screening for disease in patients. So if you're actively looking for ATTR amyloid. So we are seeing that in the clinical realm, PYP yield is much higher than in the community patients who have heart failure in whom you're looking for it. But then when you compare that, again, making a plug for PET here, when you compare that with the PET data that we have right now, there's data from Dr. Matt Moore's group, from Ahmed Masri's group, and maybe emerging data from the REVEAL study, which is a phase 3 study of evusamotide. We are seeing earlier and earlier detection of amyloid, at least in the heart, before you see abnormalities in echocardiograms or MRIs. And in fact, I can tell you anecdotally, we've had a case where Congo red staining was negative in endomyocardial biopsy, and evusamotide PET was positive. We sent it off for EM, and EM, electron microscopy, shows amyloid fibrils. So this molecular imaging techniques may be very, very sensitive to detect minute amounts of amyloid in the heart, in the whole heart, right? That's an advantage compared to endomyocardial biopsy with a small piece. And what we do with this information is the next phase of developments when we start treating them and all of that. But I think the first step is detecting them. And with AL amyloid, Sarah Cuddy's paper was shown earlier today. Early disease can be detected with F18 tracers with evusamotide as well, where you have normal echo, normal MRI, but the PET tracer is picking up amyloid in the heart well before you have other structural functional abnormalities. So the future looks very exciting.
Great conversation, Matt Moore from New York. Just one comment and then two questions. One is, if we actually based ATTR ACT on GLS, the trial would have been negative, right? There was no change in GLS, so I'm not an imager, but I warn us about getting too enamored by pictures and then finding out that, we don't have a drug that saves lives and prevents hospitalizations. So I think we need to be a little cautious sometimes and look backwards. I guess my two questions are, I agree PET is not widely available. Maybe, Spencer, you can comment. Is it possible to leverage, the peptide and use a technetium-based approach so that we can have it more widely available in clinic? And then I'm curious to hear people's comments. How early do we go, right? We know that amyloid's never normal. It's always pathologic, but are we going to find, millions of 80-year-olds with, as my grandmother said, abyssal of amyloid in their heart? Nothing's going to happen to them. Mariana's data has shown that if you have a grade 1 PYP scan, While you might have amyloid early, those patients don't have clinical events at all, nothing cardiac. And so, we're still struggling with how early to go. I don't want to ignore early disease, but I wonder what the panel thinks.
Thanks for the question, Matt. So I would argue that PET is widely available. It's just maybe not as widely available to cardiology at this point in time. So if you give a little more sharp elbows to your oncology colleagues, I think you'll find that there's a lot of PET centers in the US, including both at universities as well as private. And PET is the most fast increasing imaging modality in terms of adding new machines as well as number of PET scans. So SPECT is actually overall decreasing, not necessarily in the amyloid space, but overall. And I think, that switches, it hasn't been a big modality used in cardiology in the past, but we think certainly with the use in CAD with fluoropyridase, that is going to expand. There's a large push in cardiology, and there will be other PET tracers that are in development for other cardiology diseases. In regards to using SPECT as well with, a similar product like evazametide, we are developing something. It's an early phase, and the idea here is to kind of address what you and Prem both mentioned, which is for the places that PET may not be widely available and SPECT's more available, that you really have, potentially two tracers. One that is, for highly specialized or amyloid clinics, and one that may be more accessible to the community to get the broader perspective. And I forgot what your last question was.
So I can take that. Do you want to take that?
No, you guys go. Yeah.
Keep going if you want. I'll start with that one. So Matt always asks great questions. So regarding the GLS, I hear you, and we wouldn't, wouldn't only hang my hat on GLS, but I think GLS has tremendous prognostic value. And I think there's ample evidence in AL amyloidosis, Ashu's paper, and us also, and ATTR, growing evidence that GLS provides additive information to cardiac biomarkers and biomarker staging. Whether it changes with time or whether it stabilizes while the untreated group worsens, that's the whole point of the study, I think, to understand that. Amyloid versus amyloidosis is a fascinating question and something we talked about last weekend when we were in Italy together. And I do think a highly sensitive imaging modality like evazametide may identify people who have incidental amyloidosis that will never do anything to them. Just like, and I made the analogy last week about incidental atherosclerosis that will never do anything to them. For people who have CT scans that demonstrate atherosclerotic calcifications in their aorta or coronary arteries. We need to understand the progression of those patients. We have a very good sense about the progression and risk for people who have coronary atherosclerosis or vascular atherosclerosis by virtue of quantification of that risk, so for example, the Agatston score for coronary calcification. We may need to evolve something like that for amyloid to understand, and it may be something that has other inputs that could be age of onset or other sorts of, or sex, for example, that may predict which patients merit therapy at that stage. It's a great question.
Yeah, so Sharmila Dorbala here. Just wanted to throw in one more point, which is right now we do not know what to do with this early disease detection. Probably we did not know what to do with the calcifications, but we are modifying the risk factors, trying to kind of improve the patient's overall outcomes. If we don't look for it, we are not going to enhance the field. If we look for it and we show that it is innocuous, and if we can figure out who are the patients in whom this small amyloid doesn't progress versus who are the patients in whom it does progress, I think that would be an advance for the field and improve patient outcomes.
Well, and one thing I worry about is what happens to patients when they have a negative PYP. Are these patients getting followed, and are they ultimately get, if we all knew that they were going to get found when they were NAC stage 1, that would be one thing, but that may not necessarily be the case. a lot of times the provider may think they're negative, we're done.
There's another complicating factor, which is, and this is happening more and more lately because people are getting comfortable treating in the community. We looked at this, about now 3% of our referrals, like total referrals, are on treatment when they don't have amyloid as well. And that's based on scintigraphy, which is a huge problem. If you think about the cost of these drugs, the labels that these patients get labeled with, they actually get denied advanced therapies sometimes because they're labeled with that. I just want to ask Dr. Hoffling a question. So to move us a little bit towards the imaging and the regulatory aspects of things, I'm just going to pick on CMR and ECV for a second. So ECV, extracellular volume fraction, is meant to measure amyloid burden amongst some other things. And in a longitudinal fashion, now there is a lot of data for its prognostic value at baseline. There are emerging data for its prognostic value over change over time, and in terms of response to therapy. In your view, something like that, does that really meet, in a way, a surrogate imaging marker for actually approval processes or for approach to drug therapy in such an example? And it doesn't have to be exactly ECV, but I think, we saw people present on that.
Hi, thanks. Certainly I think that's a great start. If we just look at the, the surrogate endpoint or the accelerated approval guidance that the FDA has published, we at least want to have clinical evidence that there's a reasonable likelihood that the imaging will predict some type of clinically useful information like a treatment response. So, I'm not familiar with the data, but it sounds like, if there are data that show, clinical data particularly, that are showing prognostic value or other value that you mentioned, I think sounds like a potential starting place for validating an endpoint. Also, just wanted to, if I can, the determining whether an endpoint or imaging correlates with some clinically meaningful information, clearly that's critical. But just to go back to some comments that were made at a previous meeting of this forum, before you get there, we want to make sure that the imaging correlates with itself. In other words, it has low variance. And I may be repeating things that are very clear now, but that's also the point of this working group and the project with Echo. If an imaging study doesn't have low variance, doesn't agree with itself, it's unlikely that it's going to be useful. Even it's going to be difficult to correlate that with some type of useful clinical information. And just on that point of variance, just wanted to mention that there's a 2018 FDA guidance for industry titled Clinical Trial Imaging Endpoint Process Standards, which identifies some elements of imaging process. And by imaging process, you can think of it loosely as acquisition and interpretation, but there are many little components to that also. And if we can standardize and apply controls to those elements to minimize the variance, that's an important critical step also.
Great. Spencer, do you have a comment?
Yeah, I just wanted to kind of also address the amyloid versus amyloidosis, not from this disease's perspective, but again, looking back at Alzheimer's, which is another amyloid disease. And they have really, the clinical guidelines have changed a lot in terms of the staging of those patients. So the staging now is such that if a patient has pathology, they have the disease. Now it's considered early disease. So in their perspective, there is a clinical Alzheimer's, but amyloid itself is a stage of Alzheimer's. There's a lot of controversy around that, as you can expect, but I think where this becomes important is, they've also moved into prevention trials. And so, you know, if you see a minute trace of amyloid, are they the right patient to look at to see if you can prevent clinical disease? And I think, you know, the ACT early is a great study that's obviously being done in TTR, but I think, you know, that those kind of studies can be enriched more with early detection through, you know, potentially PET or other modalities.
So I can't speak to your elbowing cardiologists or oncologists regarding PET, but I think the other thing to keep in mind is we're talking about earlier identification, picking up what might be subclinical disease, and this is all fascinating. But the reality is so many of these patients are outside of the academic centers where these highly specialized techniques are not going to be available. I think it's important to keep us grounded on how we are going to help find those patients, because those aren't the patients we're really finding with some clinical disease. They're often way late in the process, and everything we know about the outcomes of these patients hinges on identifying them early.
As a brief, quick comment, I am actually a little more excited than Spencer seems to be about the tech case. Because, you know, if you can combine the logistic ease of use of a tech agent, as Matt was alluding to, with the performance characteristics of evuzemetide, that is a winner. I'll put some money on that. And you know, I think what you say is right. The accessibility of PET is increasing. But again, as Lori said, this is all about remote areas, you know, in the middle of the country, where I think to be able to scan thousands of patients with PET is simply not going to be possible. Now, if you have a patient with a high probability and they're PYP negative, maybe the next step is a PET. But PET as first step is going to take a very long time.
I have a soft spot for AL amyloid, so that's why, you know, I'm on the PET team, and hopefully I can convince you later that we can actually scale that based on the distribution of the non-cardiac dedicated PET scanners in the United States. So remember, we don't need a cardiac package for something like this, but I think we should just, you know, move a little bit, and we'll come back to this discussion. Justin?
Hi, Justin Groden, Dallas, Texas. So this has been a great session. Cheers to everybody that gave a talk. So I want to pivot off of one of Lori's comments and really move more towards, I think, Jeremy's talk. So, you know, with this concept of case finding, and I think it's a little bit different than early detection. So we're talking about screening or trying to identify cases where, you know, maybe the physician did not think or it didn't enter their minds. And so, you know, one issue with a number of modeling strategies, whether it's AI, whether it's linear regression, whether it's, you know, whatever the next person comes up with, they're all trained on a certain endpoint. And then when we look at these test characteristics, the performance of these models, whether it's AI, whether we're looking at ECGs, whether we're looking at an amalgam of diagnoses, perhaps some of them might even be meaningless, or imaging based with echo, the performance characteristics are all dependent on the prevalence of the disease in the population. And one of the key limitations of all of these wonderful ideas, right? I mean, really smart people are coming up with all of these techniques and all of these modeling strategies, but they all fall on that same limitation, is that the positive predictive value is quite. Now, my question, and this is, perhaps we can start with Jeremy and then maybe others on this panel chime in, is what are the right populations in which to deploy some of these techniques? Because clearly enrichment will enhance those test characteristics.
Yeah, thank you. That's a fantastic comment. You see limitation, I see job security. Well, you know, I think these are AI tools, or any tool we use in medicine is never designed to be a standalone tool, meaning two things. It should never was designed to, nor should ever screen the general population. If we want to overrun even our technetium capabilities with false positive results, that would be a great way to do it. we need to enrich, and additionally, it was never meant to diagnose disease. So these are clinical support tools, AI or otherwise. And I think, the strategy for how we enrich patients is debatable. There are probably many ways to do that. We can, utilize some of the screening studies in which they looked at older patients with HFpEF to sort of enrich that pretest probability with IWT, as you did in the SCAN-MP study. We can utilize patients with positive carpal tunnel biopsies. We can utilize even machine learning models to mine the EHR to try to identify patients at high risk that could potentially benefit from AI-guided screening. But if we're not finding some way to enrich it, and certainly we need to study whether it works in all these use cases, we're going to both lose trust and we're going to overrun the medical system with false positive AI results.
Justin, I had a comment to add. I'm sorry to interrupt. If you have an agent which is highly specific, I think the pretest probability is likely going to be less relevant. I'm not saying it won't be relevant. So that's where the molecular tracers, which directly stick to amyloid and not something else, may help.
And I agree with that point. And I also wanted to make the corollary point to your point, Justin, is that what you train your algorithm on also has to be representative of a reasonable a phenocopy, if you will. So if you train your AI on totally normal patients and amyloid patients, it's going to look really good, but it probably won't perform all that well in a test population. So I think that's one of the advantages of Jeremy's report in EHJ was that it was trained on a cohort of patients with hypertrophic cardiomyopathy and hypertensive heart disease who are rigorously phenotyped. That's what we really need. I also want to second Jeremy's point. These are decision support tools. And the last thing I'd say is that if we think about them, the positive predictive value, I'm not sure is as important. It needs to reach a certain threshold that would then lead to further testing. And so negative predictive value is also very important. Obviously, it's going to be higher in a low prevalence population, but that's another, so it's like the glass half empty, half full sort of way of looking at it.
All right, thank you. Disclaimer, I'm not a cardiologist, so neither an imager. So I'm a hematologist, but I'm just going to make two comments. One is about the grade 1 positivity for PYP TPD and what happens in the AL setting, because there is a lot of debate about how do we actually interpret it and how do we compare it and why the scan is positive. And the second bit is within PYP TPD imaging, we not only get cardiac uptake, but we also get the alteration of tracer kinetics because of soft tissue uptake. And in all of this discussion, we really haven't discussed the uptake outside the heart, which really determines what happens to the tracer kinetics. And in AL setting, we can certainly see that very different to what we see in ATTR, is there's no myocardial uptake, but there still will be soft tissue uptake and the bones can disappear. So we need to think as a group of how do we use this imaging, which is so widely used, in terms of working out the non-cardiac uptake and what the utility of that non-cardiac uptake is. And then I've got a comment, which is, again, regarding the SPECT imaging. Don't forget I-123.
Prem, did you want to answer that?
I don't know how quite you answer that. There's a lot of confusion with grade 1, whether it's grade 1 planar, whether it's grade 1 SPECT, and I think we need to do a better job of standardizing. I always say that we should get rid of this terminology because when you say grade 1, people always equate it to the Perugini score, which was planar grading. And grade 1 on a planar scan is absolutely equivocal because you don't know where the tracer is. Grade 1 on a SPECT scan is not equivocal because you know it's in the myocardium, it needs to be explained. So we have never put the grade in the report, and I'd rather call it mild SPECT uptake, avid uptake, but if it's in the myocardium, it has to be explained.
I think that's a really important point because for some of these highly specific tracers, if you're using that as a gold standard for cardiac uptake, yes or no, you'll have lots of false positives and false negatives. And I think that's really
Ashu, so I need to give you some little perspective on this. Grade 1 uptake on planar is, of course, we all agree is challenging. Grade 1 uptake on SPECT is equally challenging, in my opinion, because we do SPECT CT and I look at the SPECT only and SPECT with CT. If you see grade 1 uptake in a SPECT CT and you're confident it's on the myocardium, then we take it more seriously, obviously, and pursue further testing. But barring that, I think grade 1 uptake is somewhat of a mixed basket. And I can tell you my experience, people who are not confident in their scan interpretation, just instead of calling it negative because they're afraid of calling it negative, they just call it grade 1. So if you go with the positive negative approach that Prem is proposing, I really worry about the community. I think University of Pittsburgh can do a great job with that. But if you go out into the community, I think people are afraid of calling a scan negative, given that there's so many treatments out there. So my perspective is it's better to separate grade 1 as a separate group, negative, grade 1, and then maybe positive.
And to your point, we need to get imaging back to amyloid being a systemic disease. We essentially reduced all of the imaging to cardiology and cardiac patients, but it is a systemic disease. And that's my enthusiasm about PET, for example, is the ability to detect amyloid in most solid organs. Not all, but most solid organs with the right technique. So that's what I think that's really the big next step for us in imaging. It's not only cardiac, it's also a systemic disease.
Yeah, Ashu and I have had a lot of discussions about grade 1 recently. No, I think that's a very, very important point. And just to kind of to further reiterate what Ahmed said, the inherent quantifiability of PET. And again, to get at the previous point of we're talking about potentially early disease. Grade 1 uptake on SPECT CT is probably abnormal, but not abnormal to the point of saying they have amyloid, but we don't. So it's by the guidelines, it would be indeterminate. But I think as we understand applying therapeutics to patients with really early disease, that's where I think the inherent quantifiable ability of what we might be able to get with evazametide or PET imaging will then be able to help us provide a prediction as to whether or not, hey, you've got a little amyloid in your heart, but it doesn't matter. Just like you've got a little bit of calcium in your blood vessels, but it doesn't really matter, versus actually you're likely to progress and therefore we should treat you. We need to understand that. And we're kind of at the, I think, essentially at the very early stages of understanding that.
Yeah, I fully agree. Just one more pitch and I'll stop, which is one important point that we don't discuss is the importance of definitively excluding disease. So if a patient is in front of you with suspected amyloid, it's very important to say this patient does not have amyloid. And if you can do that with one test, right, I think that would be PET in my prediction. I don't know, but the future will tell in the next year or so. But my prediction is a highly sensitive test and a highly specific test is likely to be better at excluding disease than looking at changes in structure function or changes in SPECT traces, the mechanism of which we don't understand, right? So don't underestimate the value of excluding diagnosis.
Thank you for your patience. Please go ahead.
Sure. Christopher Gamper, Regeneron Pharmaceutical.
If you don't mind just speaking to the mic, we can't hear you.
I'll get a little closer. Chris Camper, Regeneron Pharmaceuticals. In terms of thinking about some of these imaging modalities and sort of incorporating them into clinical trials related to treatment of amyloidosis, I just wanted to see if you could comment on, and you allude to this in your echocardiography proposal, if multiple sites are participating in the study and capturing this data locally, through the local echo lab or through the local MRI scanner, how standardized are the protocols, or how could that be standardized to ensure reproducibility of the data? And do you really feel it needs a central review to ensure consistent assessment in the absence of some sort of computer-generated assessment like through AI? And a related point, on the AI algorithms, is there something that, even if it's sort of a black box, that it can illuminate back to the human observers, like what it's highlighting as features that are indicative of suspicion for amyloidosis?
Rick, do you want to start and then Jeremy after?
Sure. I mean, I definitely think that there would be a, in the context of a clinical trial, there would be an imaging protocol that would need to be followed. I think that that's standard, rigor. In the real world, I mean, the idea would be to have a readout that could be obtainable with a clinical echocardiogram that may not have followed the rigor of a clinical trial. So I think we're thinking about, in some ways, two different questions. But the challenge, I think, for us would be to not select variables that are only reproducible in the context of a specific way in which they were acquired, if that makes sense. And so that's where I think the advantage of artificial intelligence interpretation lies, because that eliminates some of the variability, obviously on the measurement side. On the acquisition side, I think there would need to be some standardization in the context of a trial. I don't know if anybody else wants to add to that.
Yeah, I can weigh in maybe on the second point, unless somebody has something to add regarding the first one. Yeah, I mean, I think that's one of the potential limitations of black box AI, right? Is the lack of explainability. There have been methods that have been put forth that to potentially look at this. You know, one of the most commonly used is GradCam maps, where you can essentially create almost a heat map superimposed upon an image, where you can get a sense of the areas that were most critical to making a prediction of positive or negative from the standpoint of the AI. It's a little bit challenging to do that because you know, it's essentially agnostic to the structure of a heart. It knows nothing about what a heart looks like or anything like that, and so it becomes a little bit challenging. And so there are ways that have actually been used to segment the heart out to into its structures and then look at the areas that become most critical. We've found, and this is a little bit more anecdotal, that it seems to be more the basal portions of the heart, like the basal LV and the mitral annulus, that seem to be the most, that light up the most.
My own AI algorithm is that if there's a lot of fat in the imaging, they don't have amyloid visually. So that's, for example, something that the AI algorithm will cheat off if you actually don't limit it to only the cardiac structure. So they cheat off where the scan was done. If there's a label on the image, they cheat off the fat content, because most of amyloid patients, 90% of them have a BMI less than 30 usually. So that's exactly the point. What are they cheating off? And you have to make it harder on them to actually cheat. Matt?
Yeah, Matt Moore. One quick comment, which is I think we're seeing tremendous number of patients with grade 1 scans. My clinical approach is to make sure I always add the word yet. You don't have amyloid yet, because I don't think we know. There are a lot of these patients who have very high pretest probabilities. Their scans are negative, including PET scans, and I never say to them, you're out of a surveillance program, because I think they could be developing it in the future. I don't know if that's right or wrong. You know, as a member of many steering committees for late phase trials, I wanted to just highlight, I've seen at least 20 or 25% of the trials have imaging that needed to be reevaluated by the core lab because it didn't work out very well. And I really want to emphasize to sponsors, I think they need to engage imaging experts early in the course of the trial and make sure if they're relying on a core lab that they know what they're doing, because honestly, I think often sometimes it's surprising core labs don't know what they're doing. And I wanted to ask Dr. Hoffling, I'm not familiar with the FDA guidance, but we heard about reproducing or reducing variability with AI. Does the FDA guidance allow the core labs to use these programs to read the images and potentially get better quality data?
Oh, I should clarify. The guidance doesn't have any specific language about AI. You asked about what, yeah, I'm not aware, at least that imaging guidance, the imaging endpoint guidance does not address AI. And I'm not exactly sure if there would be a relevant guidance from the, just also to just mention that, when we're talking about AI for imaging, this is involving software, which is regulated as a device by the Center for Devices and Radiological Health, which is not my center. But and I don't think we have. I'm not sure if we even have CDRH representatives involved in this conference, but we can definitely think about that at some point. So that guidance does not. Yeah, to sum up, it doesn't address AI. But I mean, it certainly. It sounds like AI can, and very reasonably to think that it might have a role in reducing variability. I mean, I don't see why that would not be a welcome component of a clinical trial imaging endpoint. Again, it would have to be evaluated. It would become part of the imaging process. So it would be part of the acquisition and or part of the interpretation. And we'd have to see that it minimizes variability, and then of course that it corresponds to clinically meaningful information. So not seeing why it could not be incorporated from a regulatory perspective.
It would seem like if it's FDA approved already on the market, then that would be a good usage. The other quick comment I just make about, if you will, screening or active ascertainment, one of our colleagues will present data at the AHA, but, we have an EKG screening algorithm. They identified out of like 1 million records, I don't know, a few thousand people who potentially had amyloid. And this is where I think we need implementation scientists to help us out. You pick up the phone, you've never met the patient, and you start telling them that they may have a rare life-threatening disease. Most of them hang up and don't really want to interface. And, to Jeremy's point about trust, I think we could really run into big problems if we do widespread screening. I think we need to get into, active ascertainment with a pretest probability that's reasonable. Otherwise, we're going to really disappoint, I think, people out there. Do a lot of unnecessary testing, overwhelm the system, but that's my personal belief.
Great comment. And in the last minute, I want to ask Lori one, last question. As a drug developer, how do you think about imaging yourself? Do you think of it as an intermediary thing within a trial, or do you see a future where we could actually get to a point where we're using imaging as the primary outcome for a drug, for example?
Well, I kind of punt that back to Alex. I think if the imaging modality was specific enough, reproducible, all the things that we would want, then I think from an industry perspective, we would absolutely want to consider that. Again, punting back over to Alex and FDA, it would have to meet the criteria for the agency in order for us to consider it from a clinical trial endpoint. At the end, they have that piece of it is notable and very important. So anything, Alex, from your end?
Sorry. No, I don't think I have anything to add there.
All right, Dr. Durbala, anything, or are we good to close?
We are good to close. I wanted to thank all three speakers and the discussants for an amazing discussion, and thank you all for staying. I think we now have a break for lunch. Kristen is going to tell us when to come back.